19. Drug engineering is a branch of engineering focused on discovering, formulating, and manufacturing medication as well as analytical and quality control processes. It utilizes the fields of chemical engineering, biomedical engineering, and pharmaceutical sciences. Something is covered. You remove that cover, that is called discover. So nature has lot of things in store for us to discover. We search, we find something, and then again we search, we find something better. That is called research. So today, the teachers are involved in discovery and research. And apart from their varied activities of planning, coordination, teaching, evaluation, administration, now they have taken up research. And the need of the hour is research and discovery of herbal medicine. My dear friends, on the screen, you are seeing one person. His name is Samuel Hahnemann. He is the founder of homeopathy. Actually, he is a modern physician. He is an allopath. He is an allopathic doctor. He is a practicing doctor. He had sharp observation skills. One day, in his institution notice board, he saw a notice that Jesuit priests are being felicitated. So he attended that program. The Jesuit priests who were involved in the spread of Christianity in South America were felicitated in that meeting. At the end of the meeting, Dr. Samuel Hahnemann met the Jesuit priest and then he asked them, sir, in South America, the tribals have their own religion. So in that case, how did you penetrate and spread your religion? So the Jesuit priest said, the people in South America, they suffer from a disease called malaria. In our modern medicine, we don't have any remedy for malaria. But the tribals, especially the tribal doctors, they have knowledge about a bark called cinchona. They know that the bark of cinchona is used as an anti-malarial. But this bark, this treatment is used only for the king, tribal chief and his family. So we got this data from the tribal king, the tribal doctors, and then we collected the cinchona bark and then we started distributing it to all the people who are suffering from malaria, and all of them were cured. So that is how we were able to gain their confidence. Samuel Hahnemann was very much interested about this fact. He requested the Jesuit priest for a sample of the cinchona bark. They gave him a sample of the cinchona bark. He took that bark and out of curiosity, he started tasting that bark. The bark was extremely bitter. Some people like bitter. Coffee is bitter. So by the time he walked to his room, he had consumed nearly 3 inches of the cinchona bark. He went to bed and after a few hours, he found that he started developing all the symptoms of malaria. He had fever, he had chills, he had body pain, he had ringing in the ears. He knew very well that he is not suffering from malaria. There are no mosquitoes in that area. There is nobody who is suffering from malaria in that area. He knew very well that he is not suffering from malaria. But after eating this bark, he has developed the symptoms of malaria. In his bed, he thought for a long time, what did the Jesuit priest say? A person who is having malaria, if he takes this bark, he is getting cured. Then he thought to himself, I am a person who doesn't have malaria, but I have taken this bath. I have developed the symptoms of malaria. So he thought about this for a long period of time. And the next day in the morning, he woke up, he took a piece of paper, and then he started writing, simila simile curentrum. That means like cures like. A healthy person, he takes this cinchona bath, he develops the symptoms of malaria. A malaria patient takes this bath, he becomes healthy. So, like cures like. When you cut onion, you have tears on your eyes. You have tingling sensation on your nose. When you have cold, you have the same symptoms. Tingling sensation on your nose, tears on your eyes. So, if you take onion for cold, you will be cured. So, with this phenomenon, he collected his well-wishers, and then he had selected a lot of plant drugs, mineral drugs, venoms, poisons, and he started giving it in dilute doses to the volunteers, and he started observing what are all the symptoms. This one experiment that he had done, he started his observation and inference, and he said, whoever is having these symptoms, if you take the dilute medicine, then they will be cured. And this system is called homeopathy. It has become an international system. By a simple experiment, Dr. Samuel Hahnemann was able to found a system of medicine, homeopathy. During the first wave, arsenic album was given as a prophylactic for COVID-19, for allergy, for skin disease, for diseases of the children. Today, homeopathy is a boom. All this started from a drug engineering experiment with cinchona bark. So this is called the cinchona revolution. My dear friends, we have drugs from the plant origin, we have drugs from the animals, we have drugs from the marine organisms, we have drugs from the microbial products. All these drugs have been known to us for ages. In India, the people who entered India from the Indus Valley civilization, when they entered India through the Khyber Pass, water was available in abundance. With the available water, lot of plants were cultivated. Lot of plants were available in the wild. So, concentrating on plant drugs, the people from Indus Valley civilization, the Aryans, they developed a system of medicine that is called as Ayurveda. Veda means science. Ayur means life. Ayurveda means science of life. There was a very big continent called Lemuria. This continent encompassed Sri Lanka, Malaysia, Java, Sumatra, Singapore. And in this continent, the people entered India through the southernmost tip, that is called Kanyakumari. When they entered India through Kanyakumari, water was not much available. So with the help of the available minerals and metals, they came out with a system of medicine, which we call it as Siddha system of medicine. Siddha means science. Siddha means scientist. This system was entirely written in Tamil. That is why it did not cross the borders of Tamil Nadu. Both these systems, Ayurveda and Siddha, they coexist. Documental evidence says Siddha system of medicine is 12,000 years old. So, 30 years is called as a generation. So, in the Siddha system of medicine, the knowledge on medicinal plants has been passed over generations to generations. In the first generation, it is called a home remedy. If it is successfully practiced for that 30 years, then it enters into the second generation. So then it is called a native medicine. If it is again successfully practiced as a native medicine for 30 years, then it is called as the indigenous system of medicine formulation. And then if it is successful for another 30 years, then it is called as an Indian system of medicine formulation. So 120 years of existence of a plant drug or a formulation in a community is accepted as a formulation of the Indian systems of medicine. In this 120 years, the plant drug is matured into a formulation. The dose is optimized. The plant drug might be having some side effects and toxic reactions. To counter that, they add some other drugs. Suppose if the plant is having a side effect, a dry mouth. To overcome the dry mouth, they would be adding lemon. They would be adding salt. They would be adding ginger. Now, to potentiate the activity, they would be adding some synergistic drugs. All this put together, it matures into a formulation. But the problem with this traditional formulations are they are tailor-made. That means they are made for the individual. So bulk manufacturing, as we do in modern medicine, standardization. Because at that time, this formulation, this medicine was practiced for free. They didn't charge any money. It was practiced out of passion. It was practiced out of love. So when it is practiced out of love, there is no need for adulteration. There is no need for substitution. So because adulteration and substitutions were not there, these formulations were not standardized. And it was prepared then and there and given. So no stability studies. The patient himself were taught how to prepare that formulation. So no labeling studies, no packaging studies. So these and all are the disadvantages of this traditional plant drugs. My dear friends, let me introduce you to my very good old friend, Padma Shri P. Pushpangadan. Dr. Pushpangadan was the Director General of Tropical Botanical Garden and Research Institute. When he was the director here, he wanted to collect some medicinal plants. So he started climbing the Agastya hills with his students and with his friends. When he was climbing the hill, the tribal guides who were helping him on the route, they were all very fresh and energetic even after reaching the top of the hill. But Dr. Pushpangadan was very tired. He was gasping for breath. So he asked the tribal people, how come you are all so energetic even after climbing this hill? So they said, see, in our mouth, we are having the plant part of Trichoepis xylanicus. It is called as Arogya Pacha. So when you take this plant part and if you place it in your mouth, so it is a adaptogen. So you will not feel tired. So he took the plant part from the tribal guides and he placed it in his mouth, and immediately he was able to feel very energetic. So he came down, he did research on this plant, and he has come out with a lot of beautiful phytoconstituents. He has isolated them, he has developed them, he has made them into a formulation, and he has patented them, and he has commercialized the patent, and all the money that has been generated out of this commercialization, he has given it back to the tribals. And for that, he has been given the highest award, Padma Shri. So, this Arogya Pacha is available throughout Tamil Nadu, it is available throughout Kerala, it is available throughout Andhra Pradesh, it is available throughout Karnataka, and it has been used by the tribals, it has been used in the traditional systems of medicine. but nobody came forward to do research on it. Some people have done research on this, but they just published it. But he is the one person who has done research. He has isolated the phytoconstituents, he has formulated it, he has patented it, he has commercialized the patent, and the money generated from the commercialization, he has given it back to the tribals. Dr. Marshalkar, the then CSIR director, he used to say, publish and perish, patent and cherish. So, from this one example, we clearly understand that drug engineering is something that is related to our patriotism. Modern developed countries like America, they don't have a tradition. We Indians, we should be very proud that we are having a glorious tradition. In our tradition, we have a lot of drugs. All these drugs have been thoroughly studied and they have been developed into a formulation. These formulations are available in official text. If you go through Drugs and Cosmetics Act, the official text of Ayurveda, Siddha, everything is available. If a formulation is mentioned in that official text, then it is a Indian system of medicine formulation. Now, as I said, this formulation will have some problems. They have not been studied for scale up. It has been studied only for make your medicine, tailor-made medicine. You can make 5 grams, you can make 50 grams, but you can never make 500 grams and 5 kgs, 50 kgs, 500 kgs. So that scale up studies we will have to do. Now in the modern medicine, you have different pharmacological screening methods. All these traditional plants are holistic in their treatment, but the modern pharmacological methods are not holistic. They are specific. No, to satisfy the modern world, you need to study these formulations as per modern medicine. You will have to do the scale-up studies, you will have to standardize the raw materials, you will have to standardize the in-process preparation method, you will have to standardize the finished product, you will have to do stability studies, you will have to do packaging studies, you will have to do labeling studies, and you will have to patent it, and you will have to commercialize it, and you will have to see that this money is handed over back to the tribals who have preserved this knowledge for ages. So as an Indian, it is our duty to do this. We cannot expect a foreigner to come and do research on our Indian traditional systems of medicine. My dear friends, the government of India has come out with traditional knowledge digital library. Whatever information you have in your family, your grandmother, your forefathers, you can document these information related to medicinal plants, plant drugs, plant formulations, drugs of natural origin in traditional knowledge digital library. So once it is documented in this traditional knowledge digital library, it cannot be patented. It is in public domain. So we are losing many of our plants in the name of patents to other countries. Fighting them and getting them back is very costly. So as an Indian, as a teacher, as an academician, our prime duty is to document the knowledge of traditional herbal formulations, traditional herbs in traditional knowledge digital library. I request all the active participants here to, as soon as you go home today, you sit with your grandmother, you sit with your grandfather, you try to document all the herbal formulations. What is the formulation? What is it used for? What is the dose? How do you prepare it? Is there any quality control? Is there any precautions while taking these formulations? You write all of that, and in your name, you can document it in traditional knowledge digital library. You can even document it in the name of your grandfather, grandmother who has given you this beautiful information. My dear friends, why plant drugs? One, structural diversity. Today, majority of the research is on chemicals. But though we work on chemicals, the structural diversity available in plants is very difficult to mimic. And apparently unlimited quantity. Even today, many of the herbal drugs are collected from the wild. So because they are collected from the wild, they are becoming extinct. They are becoming endangered. So we should follow good collection practices while collecting the medicinal plants. We should develop research for cultivation of medicinal plants. My dear friends, you all know CMAP, Central Institute of Medicinal Aromatic Plants in Lucknow. They have done a lot of research on cultivation of medicinal plants. Indian Council of Agricultural Research. They have done a lot of research on cultivation of medicinal plants. So we should get interested in cultivating medicinal plants. We should cultivate medicinal plants. We all know during this COVID, Andrographis paniculata, we call it in Hindi as Kalme, in Tamil as Nilavenu. So there was heavy demand for this plant. This plant is available as a weed throughout Tamil Nadu. But when the demand is suddenly increased, it was not available. So we have not developed a cultivation method for Andrographis paniculata. So now it is a need of the hour. We should cultivate it because the demand is more. And today in modern medicine, we have a lot of challenges. We have this COVID-19. By trial and error, we try to manage it with the available drugs. But when we started using steroids, we saved the patient. But what happened to the side effects? Black fungus. So for these challenges, plants as an evolution, they have answers. In modern medicine, the treatment is symptomatic. The disease can be managed. But in our traditional systems of medicine, it is aimed for cure. So the potency of herbal medicine is more. As I said, they have been documented for 120 years to enter into a system of medicine, an Indian system of medicine. So the safety has been time tested. The safety has been trusted. And the efficacy, because it is in public domain, it has been improved by addition of synergistic drugs. Like for example, you all know in most of the traditional Indian formulations, one common ingredient is pepper, piper niger. This pepper is added to most of the formulations as a bioavailability enhancer. Lot of people have studied the bioavailability enhancing property of pepper on even modern medicine. So this is the greatness of plant drugs. Now, if you look the success stories of drugs that are available in the market, 74% is only chemical drugs. 16% is biologicals. 8% is herbs. So out of this 8%, what do we have? Natural products, natural product derivatives, natural product pharmacophores, natural product mimicking products. So all these are available. Among that, we have been concentrating on antibacterials and anti-cancer drugs. Even today in modern medicine, most of the anti-cancer drugs available are from plants, vincristine, vinblastin, Nitya Kalyani, Vinca rosea. They have been successful in discovering vincristine and vinblastin from Vinca rosea, which has been used for ages in our traditional systems of medicine. I'm not able to change the next slide. Hello?
Sir?
I'm not able to change the next slide.
Use the arrow key, sir. I think it will move.
It's not moving.
Refresh
The next one, first option.
Last view. Okay, thank you.
Yes. Thank you.
Herbal medicines contain a large variety of different compounds. That is the advantage of herbal medicines. Several of these may have biological activity, but there is a significant risk of side effects and toxicity. The side effects and toxicity has been taken care in the formulation of Indian systems of medicine. But when you go back to research on an individual medicinal plant, then you will have to concentrate on the side effects and toxicity. The active principle that is present in the plant is in very, very negligible amount. So, if you purify a compound, you isolate an active compound, you purify it, the activity increases. But there are also instances where in a single plant drug, there are many compounds that work by synergism. So the more you purify it at a particular level, the efficacy comes down. Herbal medicines may be interacting with prescribed medicine. So food-drug interaction, drug interactions. So these and all are very important when we do research on herbal medicine. I will give you one simple example. We will take ginseng. If you come from Korea, the people in Korea are so patriotic, you cannot bring even a small dry part of ginseng. So in the airport, in all the seaport, wherever you try to exit Korea, the checking is so systematic and thorough that you cannot bring even a small dried plant part out of Korea. But from India, when we discovered a taxol from Taxus baccata, all the Taxus trees that were more than 8 years old, they were just cut and they were exported. Now we will come back to ginseng. So if you take ginseng, two active constituents, RG1, RB1, both are sterol ginsenosides. But one is glucocorticoid receptor, another one is a estrogen receptor. One upregulates a growth receptor, another one has a different pathway. One stimulates the blood vessel growth, another inhibits the blood vessel growth. So this is the difficulty in doing research on plant drugs. Each and every plant has different parts. Each and every part has more than 40, 50 phytoconstituents. Each and every phytoconstituent has a different activity. And the most important in this is biological variation. The same plant, like for example, we started today's lecture with cinchona. The same cinchona, if it is growing on a hilltop, it will have quinine. The same plant, you grow it in the valley, it will not have any quinine. So this is geographical variation. There is seasonal variation. The same plant, in spring season, it will have one activity. In summer, it will have another activity. In summer, some of the phytoconstituents would not be there. In rainy season, some of the constituents would not be there. So there is a seasonal variation. There is a geographical variation. There is an ontogenic variation. Based on the age of the plant, there is a changes in the phytoconstituents. So these and all are the problems faced in doing research on medicinal plants. When we do research on medicinal plants, it takes minimum 3 to 9 years to identify an active constituent and scientifically prove it. Then another 5 to 6 years, you need to develop it into a formulation. If it is not in the Indian systems of medicine, then you will have to go to clinical trials another 3 to 4 years. Then the approval process will take 1 to 2 years, and then you will have to market it. So minimum 9 years to an average of 15 years is required to bring a drug into the market. But today, time is money. So 1 minute, they say, is $1.9. 1 hour is $114. So like this, the period required for developing a herb into a drug, that time is calculated as money. So everybody is very fast. They try to develop different methods to bring it to the market. But in this research, what happens? Some of the side effects, adverse reactions are not noticed. The drugs remain in the society for 10 years, and then slowly pharmacovigilance team comes out with lot of side effects, adverse reactions, the drug is banned, the drug is withdrawn. But what will happen to the people who have consumed it in the 10 years? Now, how do we proceed in drug engineering? We develop a chemical library. From that chemical library for the particular condition, for the particular disease, we try to identify the compounds that are promising. It is called hits. And from that compounds, we try to modify it and we try to find out a lead. Then we try to elucidate the structure. Then we try to pharmacologically screen it, study it. We try to develop a dose response curve, toxicity studies. And then we go into pre-formulation studies, and then into preclinical studies, and then clinical studies, and then we call it as a discovery. Then we try to make that formulation into a patient compliant one. And for the final formulation, we go for clinical trials and then we apply for approval. So here, the knowledge about medicinal plants is very, very important. With modernization, this knowledge on medicinal plants is slowly eroding and corroding. So, documenting the traditional knowledge available is very, very important. Today, there is fruitless collection of medicinal plants. Identifying medicinal plants has become a great problem. One plant is having many names, and one name for many plants. So we will have to overcome this problem of identification. We are screening the medicinal plants today by the screening methods used by modern medicine. In modern medicine, we have chemicals. They are individual compounds. The structure is known. They are pure. But the same method cannot be used for plants. We separate a chemical with the help of HPLC. The same separation method cannot be used for plants. We have a very good library for synthetic drugs, but for plant drugs, our library is in a birthing stage. We have found a lot of lead molecules from plants, but it is only 6%. 94% are still to be explored. We have identified a lot of plant drugs, but still they are not properly utilized. New drug discovery and development from plants is going on, but if you do it from plant, as I said, it takes a lot of time, and time is money. So if you try to standardize the developed formulations from our Indian systems of medicine, then that is the smart way to do research. And the clinical trials for our traditional systems of medicine, because they are already in the community, is easy and smooth, and the approval process, marketing, everything is easy and smooth. So first and foremost, we need to identify the hits. We need to convert the hits to leads, and then we have to optimize the leads. So these are the 3 steps in which we proceed with regards to herbal drugs. Doing experiments on animals is very expensive. It causes suffering to animals. The results may be clouded by interference with other biological systems. Can you imagine? 2.6 million animals are used per year for procedures only in United Kingdom. My dear friends, but our traditional systems of medicine formulations, they have already been tested for 120 years. They are official. They are used by the Siddha physicians. They are used by Ayurvedic physicians. Only thing is, we will have to do research on modernization and make it into a new formulation, or we will have to do research on scale-up studies, standardization, stability, packaging. So these and all are the areas on which we need to concentrate, and it is a smart method it is a time-saving method, it is a money-saving method to come out, and this method is, the success ratio is very high. So, in our search for drugs, we need to use the 3 R's. We need to refine our search, we need to reduce the resources used in drug discovery, and we need to replace the modern medicine that are having side effects with those that has been time tested. As I said, the traditional system of medicine aims at cure, but today, because of modernization where nobody has time, if I have pain, I just want the pain to go away. So I will be taking a painkiller, I will be taking an analgesic. I'm not bothered about the root cause of the pain. I'm not bothered about seeing that the pain doesn't reoccur. So this mentality, this attitude has to change. My dear friends, in our traditional systems of medicine, we have a beautiful science that is called doctrine of signatures. God has put a sign on each and every plant. Only thing is we need to identify that sign. So identifying the signature put by God in nature on plants is called a doctrine of signatures. It is an organoleptic evaluation where you make use of your eyes, you make use of your nose, your tongue, your ears, and then you make use of your 7th sense in identifying the usefulness of this medicinal plant. My dear friends, you all know this kidney bean. This is a food drug. In Siddha system of medicine, they beautifully say, Unave marundu, marunde unam. So this is all a food medicine. Here food can be used as medicine and medicine is used as food. So this kidney bean, so long back in our school they used to say kidney is bean shaped. In botany class they will say bean is kidney shaped. So kidney beans actually heal and maintain kidney function. They look exactly like human kidney. So the ancient man, he was just thinking that this bean is looking like kidney. So for my ailments of kidney, if I take this bean, then I can be cured. So he started taking this kidney bean for problems of kidney, and today science has endorsed it. If you take walnut, they look like our brain. Left hemisphere, right hemisphere, upper cerebrum, lower cerebellum, everything is like our brain. And walnut improves our memory. So the ancient mind thought that this walnut is looking like our brain. If I eat it, it will be useful to my brain. And today, we all agree that walnut increases our memory. It is useful to our brain. Look at carrot. If you take a carrot, if you cut the carrot, the transverse section, in the center you can find a ring and radiating lines. They look like the pupil of the eye. So the ancient man thought that the cross section of a carrot looks like the human eye. The pupil, the iris, the radiating lines, they just look like the human eye. And the science now shows carrots really enhance blood flow to the eyes and aid in the general function of the eyes. So this is doctrine of signatures. So in nature, we have a lot of plant parts that look like our human organs. And the similarity helps them in identifying the pharmacological use of the medicinal plants. You all know about this clary. They look like the bones. And in a traditional medicine, this was not much used, but today in modern culinary, this clary is used. It has 23% sodium. So people who have less sodium, if you don't have enough sodium in your diet, the body pulls it from the bones. So the bones become weak. So foods like clary replenish the skeletal needs of the body. Avocado, it is nature's delight. They look like the womb and cervix. So it is useful to the healthy functioning of the womb and cervix because they look just like it. Avocados help women balance hormones, shed unwanted birth weight, and they deter cervical cancer. See, a look at nature, it takes exactly 9 months to grow an avocado from blossom to ripen fruit, and it's the same with human birth. Look at figs. They look like the scrotal sac. Figs are full of seeds and they hang in twos. You can find them always as a pair when they grow. Figs increase the mobility of male sperm. They increase the number of sperm as well as the overall male fertility. So these figs, they look like the scrotal sac of the male, and it is useful in overcoming male sterility. Look at mushroom. They resemble the human ear. Mushrooms have been found to improve hearing. Mushrooms is one of the few foods that contain vitamin D. During this COVID-19, people were running for mushrooms because they wanted vitamin D. Vitamin D is available freely within sunlight. You need to have a sun bath. Vitamin D is an immunostimulant. It stimulates our immunity. This vitamin D is important for healthy bones. Even the small bones in the ear that transmit sound to our brain, they need vitamin D. And it is available in mushroom. This mushroom looks like our ear. My dear friends, this is the most catchy topic during this COVID-19. In our lungs, we have a lot of branches of small airways that finish up with tiny bunches of tissue called alveoli. These structures, they resemble the bunch of grapes. They allow oxygen to pass from the lungs to the bloodstream. A diet high in fresh fruits, such as grapes, has been shown to reduce the risk of lung cancer, emphysema. Grape seeds also contain a chemical called proanthocyanidin, which reduces the severity of asthma triggered by allergy. And these grapes, they are prophylactic for COVID-19. They are useful in the management of COVID-19. So during this COVID-19, there was one more discovery in this doctrine of signatures. That is, the coronavirus looks just like our pepper, black pepper. So we have Piper nigrum fruits. We call it as pepper. In Tamil, we call it as milagu. The coronavirus just looks just like this pepper. And if you go through our traditional systems of medicine, Siddha systems of medicine, it is said that this pepper, which resembles coronavirus, is useful in the management of all the symptoms caused by coronavirus. So if you take pepper, you can escape from the attack of coronavirus. Ginger, it looks like our stomach. It aids in digestion. The Chinese have been using it for 2000 years to calm the stomach, to cure nausea. And it is a common remedy for motion sickness. If you go to our bus stand, you can see people selling inji marappa. So it is inji maram manam tarum padu, manapadu, and that has become marappa. We also have a Unani formulation, murabba. So this ginger, the outer skin is peeled because it is an underground modification of the stem. Because it is from the ground, on the outer skin, you can have some harmful microorganisms. So the outer skin is peeled and the fresh ginger is considered. If you make it into small pieces, and if it is just sprinkled with lime water, and if it is dried, It is used as dry ginger. We call it as sukku. So in Tamil, we have a beautiful proverb. Kalayil inji, Nanpagal sukku, Malayil kadukka, Mandalam undu vandal, Kolai mundri nadakum kiravanam, Kolai visi nadapane. So, in our traditional systems of medicine, it is said that in the morning, you will have to take ginger. During this COVID-19, I was able to document that around 30% of the people in Pondicherry in the early morning, they were habituated to take ginger. And the people were very intelligent. Just before lunch, the first ball of food they used to take for lunch, kavalam, we call it in Tamil. They used to add sukku podi, there is dry ginger powder, they used to make it into a ball and then they used to consume it first. So, the usefulness of ginger was appreciated by almost 30% of the population during this COVID era. Look at sweet potato. They look like our pancreas. And they actually balance the glycemic index of diabetics. Look at olives. They assist and help in the health and function of ovaries. They look like our ovary, they help in the healthy functioning of our ovary. And that is why people are behind olive oil. So all this put together is called the doctrine of signatures. So this is our traditional systems of medicine. So the ancient man was able to identify drugs based on their resemblance with human organs. And these drugs are available in a particular season. But we have disease throughout the year. So people started developing methods to preserve the drugs of nature in yesteryears. The first method was drying. If they don't dry and preserve it, there is growth of fungus, there is growth of microorganisms, there is decay, there is action of enzymes. So they started drying it. Again, drying it in the sun, drying it in the shade, drying it by artificial heat. And then they developed the method of extraction. Maybe the first drug that they stored, it was drenched in rain. The water that came out was found to have the same activity. So they developed extraction. Initially aqueous extract, we call it as decoction. The coffee that we take today is a decoction. So in our Siddha system of medicine, we have Kudineer. Today for this COVID-19, almost 80% of the Tamilians, they had taken one simple medicine called Kaba Jwara Kudineer. When we had chikungunya, the same 80% of the population of Tamil Nadu, they have Nila Vembu Kudineer. So Kudineer is a decoction. So in this Kaba Jwara Kudineer, we have a polyherbal formulation, Kabam, sputum, Jwaram, fever, Kudineer. So when we have sputum and when we have fever, and that is related to viral fever and that was related to COVID-19, so you will have to take this formulation, you will have to tailor make it. So you will have to take the churna. It is a solid dosage form where the individual drugs are powdered and then they are mixed. And this churna is taken, it is added to water and it is boiled and then it is reduced to 1/8 and then it is filtered, the mark is pressed and the filtrate is taken and then it is consumed as such. So this is called the Kudineer. So these Kudineers are our traditional systems of medicine formulations. But the problem with Kudineer is it has a shelf life of only 3 hours. You will have to consume it within 3 hours. And every time you will have to prepare it afresh. So now in this modern world, nobody has time for it. So people have started modernizing this Kudineer formulation. They make an aqueous extract, they spray dry it, they make it into a powder, it is made into a sachet. And then you just open that sachet, you put it in water, you just stir it and then you drink it warm. So this is called modernization. But this modernization should not hamper the efficacy of that traditional formulation. So in that we should be very, very careful. My dear friends, we all know that the world's first university is Jayaseelan, started 700 years before the birth of Jesus Christ. The world's second university is also in India, Nalanda, 400 years before the birth of Jesus Christ. India was the first to give to the world the number 0. The value of pi, 22/7, was first given by Indians to the world. The first source of diamonds is India. The first cesarean operation was performed by Shushruta. The first operation for glaucoma, Shushruta. The first operation for removing kidney stones, Shushruta. The first operation for reconstruction of nose, Shushruta. Wireless communication, radio, was first invented by Professor Jagdish Chandra Bose 5 years before Marconi. Chess was invented in India. Algebra, trigonometry, everything from India. So we have a very, very glorious future. And all what our forefathers did was science. And for 500 years, we were as slaves under foreign rulers. 300 years under the Mughals, 200 years under the British. So 500 years of slavery, we have lost our traditional wealth, and we have lost the confidence that we had on our traditional medicinal plants. Now, as teachers, as teacher researchers, it is our duty to bring back this confidence that we had on our medicinal plants. My dear friends, in our Siddha system of medicine, it is beautifully said, if you are a human being, you can suffer from 4,448 diseases. Even with the help of modern technology, it is very difficult to come to a number 4,448. In the Siddha system of medicine, the diagnosis for this 4,448 disease has been clearly given. 2/3 of the Siddha system of medicine is dedicated only for preventing this 4,448 disease. Prophylaxis, how to prevent this 4,448 disease. That is very important. And then 1/3 of Siddha system of medicine is devoted for cure for this 4,448 disease. So such a beautiful system of medicine is the Siddha system of medicine. It is completely developed, completely mature. The government of India is giving lot of emphasis for Ayush. Ayush is Ayurveda, Yoga, Unani, Siddha, and Homeopathy. The S in Ayush is common for Siddha and Sauripa. So all these systems of medicine are considered as Indian systems of medicine. But when you go into history, only Ayurveda and Siddha are truly Indian systems of medicine. When we were under the Mughals, we received the Unani system of medicine. From German, we adopted homeopathy system of medicine. There was one great saint near Chidambaram. He was called Patanjali. He came out with the yoga system of medicine. And we have a way of life. The way of life of the Tamilians and of the Indians is called as naturopathy. So we have Amchi system of medicine, which they practice in Ladakh. All these are also Indian systems of medicine. In Ayurveda, it is beautifully said, Amantaram Aksharam Nasti, Nasti Moolam Aushadam. That means, in the Indian language, Sanskrit, there is no letter without a magic spell. Amantaram Aksharam Nasti. Nasti means no. Aksharam means letter. Amantram means, mantram means magic spell. Amantram means without magic spell. So without magic spell, there is no letter. Nasti moolam aushadam. Aushadam means medicine. Moolam means root. Nasti means no. There is no root without medicinal property. This is the meaning. What a beautiful meaning. You know, there was a recent beautiful research in yoga. When you have dry mouth, usually nowadays we drink less water, and when we take many modern medicine, one of the common side effects that we get is dry mouth. So for dry mouth, if you say these two letters ish na together continuously, so when you say ish, then tongue massages the sublingual gland and there is secretion of saliva. So ish na. When you say, you can see that there is flow of saliva in your mouth. And they say that is why Indians had a habit of saying Ishna in the name of Krishna. There is nothing religious about it. Don't make everything religion. The entire Ayurveda, the entire Siddha has been made into a religion. Just for the purpose of religion, we can't avoid these herbal drugs. You go to Shiva temple, they will give you bay leaf. You go to Vishnu temple, they will give you Tulasi leaf. You go to Mariamman temple, they will give you neem leaf. You go to Ganapati temple, they will give you Arugambulu leaf. So each and every plant has been made divine. If we had not tied a yellow cloth on the neem trees of Tamil Nadu, we would not have even a single neem tree. Today, neem is a green gold. It is the best natural pesticide that is available. And it is the most safest pesticide available. So in agriculture, most of the organic pesticides are neem based. You know, in Andra Pradesh, how do they celebrate the New Year? They celebrate the New Year, Yugadi, by taking the flowers of neem, the tender twigs of neem. So Yugadi comes during the flowering season of neem. So during Yugadi, there is a seasonal change. Most of the people during Yugadi celebrations will have cold, cough, allergy, and for that, the prophylaxis, the management, the cure is the flowers of neem. So that is why it is called as Yugadi Pachadi. So people who speak Telugu, they celebrate the New Year with a bitter aqueous decoction of neem flowers, mango, jaggery. So this is a formulation, time-tested formulation, that is a prophylaxis. Now, have we standardized that formulation? How much neem flower should be there? What dose we should take? What is the stability? So that we have not done. If we don't do it, after a few years, this formulation would have been gone away from our community. My dear friends, you all know about this plantain tree. Any function in Tamil Nadu will be incomplete without tying this plantain tree in front of the house. Why did this habit come? In those days, the women and men in the home, they used to cook. And it was a joint family. So minimum 40 people in one home. So if it is a function, then it is 400 people. So the people who have been trained to cook for 40 people will now have to cook for 400 people. So when they do the scale-up cooking, there are chances there could be a fire accident, there could be a burn. The first aid for burn is the fresh juice from the plantain pith. From this banana plant, if you take the center stem, that is called pith, if you squeeze it, you will get a juice. That juice is used as a first aid for burns. So that is why when there is a family function, this plantain tree was considered as a first aid box. And it was tied on the doors. It has an aesthetic appearance. It is beautiful. That is separate. But when you cook, when there is a function, there could be a burn. There could be a cut. There could be a nick. For all that, the first aid is the fresh juice of plantain pith. So that is why we have been using this plantain tree. But today what has happened? Instead of this plantain tree, they put a serial light of the image of the plantain tree. So it has become a custom. So the science behind this custom is lost. This is science. So we need to prove that the people were using the fresh juice of the stem of this plantain tree as a first aid for burns. You have to do research on the fresh juice, on its stability, on making it into a formulation. So that is the patriotic duty and responsibility of each and every Indian. In India, after food, we had a habit of consuming betel leaf. This betel leaf is an excellent digestant. Research on betel nut said that it could be a cause for cancer. So that research has made us stop this habit of having betel leaf after food. You know, this betel leaf, especially the leaf tip and the petiole, they decay first. When you cut this leaf, the cut petiole will immediately become brown in color because of enzymatic action. So when they consume this betel leaf, they remove the apex of the leaf, they remove the petiole, and then they put this leaf upside down. Under the back of the leaf, they smear it with calcium. We call it in Tamil as sunnambu. Because on the back side of the leaf, some small insects might have laid their egg. So to kill the small insects and their eggs, they smear it with lime, and then they roll it, they put it in their mouth. The first saliva, they spit it out. The second saliva, they swallow. So the method of taking a betel leaf has been standardized. This betel leaf is an excellent digestion. Today we consume a lot of food without taking a digestant. Most of them do not get digested. They create havoc to our digestive system, and they are the root cause for a number of diseases that we suffer today. In yesteryears, we were always traveling from unknown to known. We were traveling from known to known. We were never traveling from known to unknown. Here you are able to see one beetle. This is called rhinoceros beetle. This beetle, when it flies and sits on the shoot tip of coconut tree, they start eating the tender shoots of the coconut tree, and slowly the tree will not yield coconuts. To overcome this, in our modern agricultural method, they drill a hole in the stem of the coconut tree, and then they inject a substance. This substance travels to the shoot tip, and when this rhinoceros beetle eats the shoot tips, it dies. But the coconuts that comes from this becomes poisonous, not edible. So you'll have to wait for 6 months for the drugs to completely go away from the plant. On the contrary, in our traditional systems of medicine, our forefathers have studied the body structure of this rhinoceros beetle. Even with a knife, you cannot cut the body of this beetle because the skin is very thick. You bury it, it will not die. You put it in water, it will not die. So they studied the morphology of this beetle, and in between the head and the body, the neck is very soft. So we used to have some people who used to climb the coconut trees. That community was called as Sanan. So those people, to overcome these beetle nuts, they used to take sand. They used to sieve it in a 60 mesh sieve, and that fine sand, they used to take it and put it on the coconut shoot tip. So when this beetle goes to the top, and when it tries to eat the the coconut shoot tip, the fine sand goes in between the head and the body, and this sand causes friction when it is eating, and there is a wound and there is a sepsis, and this rhinoceros beetle die. So this is a mechanical method of removing this rhinoceros beetle, where no harm is done to the plant, and the plant fruits and the products can be used immediately. So this is going from unknown to known and known to known. But today, in the name of science, we are making our own food substance as poisonous. I will give you one example. This is fridge. But today, this fridge is harming our body. I went to one of my friend's home. I opened their fridge, and in that it was written Monday, Tuesday, Wednesday, Thursday, Friday, Saturday. I asked this, what is this? He said, we husband and wife, we both are going to job, so we cook only on Sunday. So you can see this is Monday idli mavu. The same idli batter on Tuesday, we used to put some onions and make it into dosa. And this is the chapati flour for Wednesday. And this is for Thursday. We have ada mavu. Ada is South Indian preparation. And on Friday, whatever is left out, we put it together and then we make it into a mixed dosa. So this is what a fridge is used for? Stale food is not healthy. This fridge was invented for a different purpose. Now it is serving a different purpose. Convenience has overcome health. My dear friends, if you go to the market, if you buy cabbage, you just smell the outer layer, you can find the smell of pesticide. You take onion, you remove the outer skin, you can find some black powder, mold. You take grapes, on the outer you can see some white coating, that is pesticide. You take apple, the outer surface of apple you just scrap, you will have petroleum jelly, apple polishing. You take greens from the market that is available, you put it in water, the water becomes green in color. Because to enhance the color, they add malachite green. You go to the nearby shop and you buy cuscus, you smell it, you have the smell of pesticide. Jaggery, they add urea to enhance the color. Even for making idli batter. The other day we made a small research. In Pondicherry they sell idli batter, idli mavu. They start selling this idli batter from afternoon 3 o'clock till night 10 o'clock. And this idli batter doesn't ferment. Then we did research how it doesn't ferment. So we found that the people are adding boric acid to the idli batter to retard fermentation. Boric acid is okay, but taking boric acid daily, it will be harmful. Who has permitted them? Just for the sake of convenience, they have added this. And what will happen to the people who are taking it? So these and all are some of the questions that we teacher researchers will have to work on. We did one research on cardamom. 90% of the cardamom available in the local market in Pondicherry, they have been coated with a coloring material to enhance the appearance. But when we cook, this cardamom, we take it in many places along with the outer skin. They just take the cardamom, they put it in mixie, they add some sugar and then they grind it, and then they use it in making sweets. So the outer skin of cardamom is having unapproved dye coated to enhance its appearance, and these dyes are carcinogenic, that is cancer toxin. Recently we did one research on the coconut chutney available in the hotels in Pondicherry. Most of the hotels, they don't use fresh coconut. They use desiccated coconut, and this coconut desiccated is from coconuts from which coconut oil has been removed, and the mark is converted into desiccated coconut for making coconut chutney, made to look like fresh coconut in local hotels. So all this are harmful to our health, and they are made intentionally for the purpose of convenience. And if we don't concentrate on doing research and creating awareness on these aspects, our health will be jeopardized. My dear friends, we all know about turmeric. Turmeric has been used for ages in India. Any auspicious function, we make use of turmeric. And turmeric has a beneficial effect on almost all parts of our human body. Brain, lungs, stomach, kidney, intestine, large intestine, small intestine, liver, heart, you name it, it has a beneficial effect. And the two Indians, they went to America and they have patented this turmeric against India for America. And we had to spend so much of money to get back this patent of turmeric. So where has our patriotism gone? So instead of publishing, we should go for patenting. And the problem is with the documentation. It was Dr. Marshallkar who fought and got back the patent of turmeric. But to get a document to prove that we have been using turmeric for so many conditions for ages, where is the documental proof? That was not available. To collect it, it was very difficult. See, you take any auspicious function in India, we make use of turmeric. Even the letter in those days, marriage invitation, an auspicious function invitation. On that letter, we used to smear turmeric and send, because through that letter, microorganisms should not travel to harmful, pathogenic microorganisms should not travel. When there is a function, what do? They mix turmeric with rice. In Tamil, we call it as akshadai, and it is sprinkled so that the air is purified of harmful microorganisms. Each and every woman, even men in those days, they used to take this turmeric, they used to add a lime, sunam. So it used to become red in color. That was called as kumkum. So that was placed on the forehead. So there is a sustained release of curcumin in the skin. But this kumkum was made as a religion. It was said it is only for Hindus. So now people have stopped wearing kumkum, which we have been wearing for ages. So in those days, they used to take this turmeric and they used to smear it on the stove so that it wards off insects. Even in a death house, when you come from a death house, they used to take turmeric and they used to sprinkle on themselves. In a birth house, they used to sprinkle turmeric. In a puberty function, it's called in Tamil as manjal neer. In temples, disinfection is with the help of turmeric. And at the last, in Tamil, the people who are 40 plus, they used to end their food with golden milk. We call it as tangapal. In the night, 2 hours before going to bed, after the dinner, after the supper, they used to take milk. They used to add turmeric. It used to become golden yellow color. And then they used to add garlic pearls. They used to boil it in the milk and they used to take it. So this turmeric with milk is an excellent antibiotic, antimicrobial, anti-ulcer, antioxidant, and the garlic, it will monitor the bad cholesterol and it is good for our health. My dear friends, you can see these are all of the diseases, conditions for which turmeric is used. The active constituent of turmeric, it is called as curcumin. It is having excellent activity against psoriasis and vitiligo. That is why the women, when they go for bath, they used to take the combs of curcuma longa. They used to scrub it on the stone with water, and the fresh paste, they used to apply it all over their skin. In all food substances, we used to add turmeric because it is prophylaxis for diabetes. It is prophylaxis for diabetic microangiopathy. It is prophylaxis for diabetic nephropathy. Even for cardiovascular disease, acute coronary syndrome, atherosclerosis, for a number of diseases, neurodegenerative diseases, for cancer, a variety of cancer, for viral diseases, for inflammatory diseases, for all this, you go through the literature. Curcumin has such a beneficial activity and they have been using it for ages. But now we are away from curcumin. So we need to popularize the use of curcumin. My dear friends, I think I will use this slide as the last slide. I have still a lot of slides to go. The time is already 11:10 and we will be also having a question and answer session. In Tamil Nadu, 40 years back, the Tamilians, they used to break their fast with coffee. They used to have their dinner or supper in the night before sunset. And next day morning, they have been fasting for nearly 18 hours. in the morning, they break their fast with coffee. The coffee that the ancient Tamilians took was 65% chicory, 35% coffee. What is this chicory? Wherever red soil is available, down south, that is, you can take Thoothukudi district. So in all that place, you have chicory. In Tamil, we call it as chicory. In English, we call it as chicory. The binomial nomenclature is chicory incubus. So it is an underground root. They uproot the chicory roots. They cut it into small pieces. They remove the outer skin. They dry it and then they powder it. So this is called as chicory powder. 65% chicory, 35% coffee. Why coffee? Because this chicory is bland. It doesn't have any taste. And so the taste is not proper with chicory. So to enhance the taste, they used to add coffee. What is coffee? Coffee is from plant Coffea arabica. So it has a one-seeded berry fruit. You allow the fruit to mature, and then you wash it in water. You get the seed. You dry the seed. You roast the seed. You powder the seed. That is called coffee powder, and that one you mix it with chicory. Why chicory? When you take chicory in the early morning on empty stomach, it has maximum amount of soluble fiber. So the soluble fiber in chicory, they will clean our intestine. That is why chicory was taken in the early morning, number one. Number two, this chicory has a product called inulin. This inulin is a delicacy for microorganisms. So the microorganisms in our stomach, the good microorganisms in our stomach that help in our digestion, they love to eat inulin. This inulin is present in chicory. It's present in the middle lamella of chicory, and when you take this chicory powder, so the microorganisms in our stomach, they feed on this inulin, they become healthy, and throughout the day they will be helping us in digestion. That is why they were taking this chicory powder with coffee in the morning. Now, because of development of science, we thought coffee, because it has caffeine, and caffeine because it is a central nervous system stimulant, we are taking coffee in the early morning. So this chicory was not having caffeine. So this chicory was branded as an adulterant, as a substitute. So they said you should not mix chicory with coffee. We thought that this is a good science, and slowly the percentage of chicory in coffee got reduced. It became 50-50, and then it became 65% coffee, 35% chicory, 90% coffee, 10% chicory, and now no chicory, pure coffee, and then now no coffee, pure granulated caffeine. So once we were taking only chicory, because the taste of chicory was not nice, we added coffee. Now we are taking only coffee and we are not taking chicory. And abroad, if you go to America, most of the people there who are well educated, who are health conscious, they start their day with inulin powder, with organic chicory root powder. And this chicory is also popular in Unani system of medicine. It is called as Casinipide. Now, what are all the health benefits of inulin? It aids in calcium absorption. It improves good cholesterol. It increases the number of good bacteria in the gut. It helps you feel fuller for long. So these are all of the benefits of inulin. And what does the soluble fiber in chicory do? They prolong the stomach emptying time so that sugar is released and absorbed more slowly and make you full for long. They lower the total cholesterol and low density lipoprotein cholesterol. There is a bad cholesterol. Therefore, the risk of heart attack is reduced. It reduces blood sugar. The other sources is oat, soybean, peas, nuts, barley, fruits, and vegetables. But among all this, chicory has the maximum amount of soluble fiber and maximum amount of inulin. So having chicory in the morning is best. But today, most of the coffee that we take is devoid of chicory. Okay, see, I have a lot of other slides, but because of paucity of time, I will not touch much. This is our Nilavembu. Andra Pradesh paniculata. We call it as Nilavendu Kudinir. During the period of chikungunya, Ebola, this was a very good drug that we had used. And it was a make your own medicine type. So it has to be prepared fresh. This has to be standardized. My dear friends, this you might have seen in most of the houses in South India. In the entrance of the house, they try one sulfur ore, one lemon, chilies, and then one thorn, one lemon, and coconut. So this was a first aid box of yesteryears. The first one, sulfur ore, is a air purifier. The second one, lemon, in case of poisonous bites, they used to have dehydration. So for that it was used. Again, the oleoresin of chili was used for poison bites. The thorn was used for removing the venomous animal or reptile parts that are stings in our body. And turmeric, antiseptic, and coconut is anti-hypoglycemic. So this started as a first aid box of yesteryears, but still we have not popularized it. We have not done research on it. In yesteryears, the medicine that were tailor-made, it was free medicine. We were making use of the use and throw toothbrush. In Tamil, we used to call this Alum Velum Pallukku Urudi Nalum Rendum Sollukku Urudi. So meshwak, neem, these and all are the twigs that were used as toothbrush. They were use and throw. We were using charcoal, the best absorbent. We were using salt as toothpaste. But now we had gone to modern paste. And today, even in the modern paste, in the advertisement, you can see they are asking whether your paste is having salt, whether your paste is having charcoal. So it is one cycle. In the name of modernization, we forget our tradition, and again we go back to our roots. Every season has a fruit, and these fruits, they help us to overcome the mineral deficiency, the vitamin deficiency that we suffer. Zinc, when you take it in an organic form from fruits and vegetables, they don't harm you. Lemon juice, fresh lemon juice. Today, in most of the hotels, instead of fresh lemon juice, they use of synthetic flavors and enhancers. Lemon is easily available. The skin of lemon is also having medicinal property. Lemon, when it is immature, it is green. When it matures, it becomes yellow. When it dries, it becomes brown. So that is the greatness of this beautiful fruit. But in the name of modernization, we tend to add artificial flavors, sweeteners, and so the original concept of using it as a medicine is lost. Chyawanprash, it has 46 ingredients. Main one is amla. It is popular for its vitamin C content. Vitamin C is stable when it is in a lipid form. Now the same is available as a biscuit. And this biscuit doesn't have the lipid content, so here in this biscuit, the vitamin C is lost. So in the name of modernization, we are losing the reason why we went into this concept of having a traditional formulation. My dear friends, even the food that we eat today is adulterated. To detect this adulterated food, as I said, in our traditional system of medicine, food is medicine and medicine is food. Milk is used as a medicine, it's a vehicle for medicine in our traditional systems of medicine. And this milk today is mostly adulterated. So to find out this adulteration, the government of India, FSSAI, has come out with a DART, detect adulterants with a rapid test. So similarly, we will have to come out with methods to detect adulterants of food medicine, of herbs, herbal products. So this is a very lengthy topic. Maybe next time when I have time, we will go into it. To sum up, I humbly request each and every participants who have been active throughout this session to document the medicinal properties of plants, to document the medicinal properties of the herbal formulations, natural formulations, household formulation, home remedies that have been known to them in traditional knowledge digital library. Number one. Number two, take one formulation. Do research on their formulation. Please don't publish, patent it. Commercialize that patent and make use of the patent for the development of our traditional systems of medicine. If we do not standardize our traditional systems of medicine formulations, these formulations will be lost forever. I was saying about this Nilavembu Kudineer. This Nilavembu Kudineer is available throughout. It is now become a cottage industry. In every house they make this Kudineer. If it has to have some 15 ingredients. If one ingredient is not available, it is conveniently forgotten. We do not have an analytical method to study if all the 15 ingredients are present in Nilavembu Kudineer. Because each and every ingredient has around some 40 compounds. If you select one marker compound, it may be not fruitful. You have to go for multiple markers. Now, establishing analytical method for this multiple markers and stabilizing the formulation is a Herculean task. But that task we will have to do it because it is our duty, our responsibility. We cannot anticipate any foreigner to come and do research on our traditional systems of medicine. So with this humble request, I once again thank the organizers for their wonderful organization, and I thank the active participation for their very active involvement in this session. Thank you one and all.
Thank you so much, sir, for your more informative and excellent talk on plant-based engineering, which is a need of our in this pandemic situation. Dear participants, if you have any queries with our resource person, you can unmute your mic and you can speak, or else you can type in the chat box. Thank you. Any queries from the participants? So on behalf of our management, deans, principal, organizing committee team, we heartfelt thanks to our resource person, Dr. B. Gopal sir, for this wonderful and excellent information session. Thank you so much, sir. And sir, one question which was asked by Dr. Vijay. What are the reasons that Indian traditional medicines are so worked out as CCMs?
Okay.
Traditional Chinese medicine.
Okay. Thank you very much for this nice question, Dr. Vijay Paramanik sir. The Chinese are very patriotic. If we go to China, we got some information that around 90% of the Chinese, they still rely on the traditional systems of medicine. They religiously follow their traditional Chinese systems of medicine. And the patriotism has helped them a lot, that the Chinese system of medicine has developed so much. Our Indian traditional systems of medicine, be it Ayurveda or Siddha, are still far lagging behind than the growth compared to Chinese systems of medicine. Number one is they don't have research, that is, we don't have a research policy. Like, what are we aiming at? Where should we be? And what time we should reach that target? So this research policy individually for the systems of medicine are still on the anvil. Awareness among the general public and the systematic scientific research, the confidence level on our traditional systems of medicine. So these and all are at a low ebb. After this COVID-19, people have started realizing the usefulness of our traditional systems of medicine, but still we tend to do research on a foreign topic, on a topic on which the foreign community is at interest rather than doing research on our own traditional systems of medicine. So we need to be more patriotic. We need to take steps. We need to create awareness. We need to become fans of our traditional systems of medicine. We need to follow this traditional systems of medicine. And if we do this, it has to become a mass movement. If it is done, then we would be competing with Chinese systems of medicine. The Chinese systems of medicine, the Korean systems of medicine, even the African traditional systems of medicine, all of them are developing very well. We are also developing, but still, if you look at the monographs of our traditional systems of medicine, Ayurvedic monograph, Siddha monograph, still we need to go a long way up. Our regulatory authorities, still they have not come out with methods of detecting adulterants, substitutes. Still, we are not very confident about the authentication of the raw materials that we use. Because in the name of money, the authenticity, the purity of our traditional formulations is at stake. In the name of modernization, we are losing the active principles of many compounds. Please don't quote me. We came across a number of Ashoka bark samples that were not actually Ashoka bark, but sal from the plant sal. So just for the purpose of money, people have taken the bark of sal, they have powdered it, they have labeled it as Ashoka bark, and they have supplied it to pharma companies. And pharma companies have prepared Ashavas and Aristas and cardiotonics based on this pseudo Ashoka bark powder. So now they will not have any effect. Then the people will lose their faith in Ashoka bark, and slowly Ashoka bark will move away from our traditional systems of medicine. So this quick money, fast money, compromising on our trustworthiness, all this have jeopardized the growth of our traditional systems of medicine. We can overcome all this by our patriotism. Thank you. Hello?
Sir, one of the participants has raised the question as, what is the
okay.
Sir, are we audible, sir?
Yeah, you're audible.
What is the scope of quality control and standardization of herbal drugs in India?
Excellent question. Thank you very much, Mahisha K. See, the need of the hour is standardization of herbal drugs. But today, we are attempting to standardize the herbal drugs just like standardizing modern medicine. So that will not work. First of all, we will have to standardize the method of authentication. Then we will have to standardize the good collection practices. Then we will have to standardize the good agricultural practices. And then we will have to come out with a monograph for individual plant drugs. Then we will have to come out with quality concepts for in-process quality control. The final finished product quality parameters will have to be worked out. And so this is what has to be done. So if you look, I think we have around 1 lakh formulations that are official. So in that only 6% that have been quality control studies have been done. So a lot of work is still pending. We will have to take interest in it and we will have to do it. So it is only the involvement of all the science community of India will help us to overcome and establish the quality parameters of herbal drugs in India. That will be a massive boom for the herbal drug market in India abroad. Thank you. And I think I have a beautiful question from Dr. Puniti Mathu. Synergy of active ingredients from a medicinal plant versus one active ingredient from that plant as a drug. Would you like to comment, please, keeping the regulatory approvals in mind? As I said, if you take a plant, if you take an active ingredient, if you isolate that ingredient, if you go for structural elucidation, and then if you try to do a pharmacological screening, toxicological studies, and if you try to market it, it is considered as a new drug. So if it is taken as a new drug, then you will have to undergo all the preclinical and the clinical phase 1, phase 2, phase 3, phase 4 trials, just as you do for modern medicine, which is time consuming and it takes a lot of money. On the contrary, as I have said, we have official formulations. Official formulations means formulations that are official in Ayurveda Pharmacopoeia, formulations that are official in Siddha Pharmacopoeia. So if you take any such one formulation and if you standardize it, in that formulation, each and every herb is made to work with synergism. So there the results would be better. So there we will be able to bypass phase 1 trials, and there we will be saving money. And there, because it is our own traditional systems of medicine which has been tested over time, which have been trusted. So if we do research on it, so as I said, no, Nilavembu Kudinir, it has 15 ingredients, but the active ingredient is only one. The all others, they work on synergism. They increase the bioavailability, they reduce the side effects. So going for such methods is far better. So in yesteryears, they were taking the whole plant. Then they came out with a particular part. And then from that part, they went to an extract. From that extract, they have come out to a compound. Now this compound is not as effective as the original plant. Now it is the other way around. From the pure compound, they want to go to an extract. And slowly from the extract, they will go to the part. And from the part, they will go to the plant. So this is one cycle. So thank you for this nice question and for your involvement. We have one Dr. Arun P. Who has asked a beautiful question about Dr. Anandaya from AP, who has prepared a herbal medicine for COVID-19 treatment. What is the current status of the medicine? So we had a beautiful team consisting of traditional doctors, physicians from Ayurveda, and we also had the scientists from Tirumala Tirupati Devasthanam institutions, and both of them as a committee, they have studied the formulation, they have gone through the ingredients. All the ingredients are safe, all the ingredients are official, all the ingredients have been used in various forms in the treatment of various conditions. So only the formulation is different, but the raw materials are already official. Hence they have permitted the use of this formulation. Now this formulation will be taken up by Apex Research Laboratories, where it will be standardized for its raw materials, in process and finished products, and it will be slowly made into a formulation, standardized formulation with stability studies, dose response curve, and toxicity studies, and it will be made into an official formulation. Thank you. Which medicine is standard medicine for cancer among homeopathy, Siddha, Imani, and chemotherapy, etc.? There is any specific compound used for treatment of cancer, sir? So I am very much thankful to Mr. Niranjan Kumar for this beautiful question. See, there are many types of cancer. There are many stages of cancer. And again, the individual health level is very important when we deal about cancer. So we cannot say one medicine. Each and every system of medicine has a number of remedies for cancer. As I said, vincristine is there, vinblastine is there. You take any plant available, the plant will be having antioxidant activity. That plant which has more antioxidant activity, they will be also having anti-cancer activity. So we need to tailor make it for the particular disease, for the particular stage, for the particular individual, and then in a particular dose, in a particular time, and then it will be effective. So generalized, we cannot say that this is the best cancer drug. That is not a right way of saying it. All plants have anti-cancer activity. But in the name of plant, we cannot go for as I said, if you take kaskasa, opium poppy, if you buy it from the market, if you smell it, you can find pesticide smell in that. Somehow they add this pesticide to see that no micro growth of insects, pests or microorganisms is there. Now this pesticide residue is itself carcinogenic, it will cause cancer. So we need to do a lot of research in this area, and we cannot generalize. Thank you very much, Mr. Niranjan Kumar. And we have the last question from Mr. Satish Raut. For golden milk, which kind of turmeric will be good? Raw turmeric, powder turmeric, or curcumin ingredient? It is powder turmeric. Raw turmeric you need to boil for a long period of time. So powder turmeric is the best one for golden milk. Thank you very much for your involvement in this session. Thank you.
Thank you so much, sir, and thank you for explanations for the questions which was raised by our participant. It's an honor for us to be here with you. Thank you so much, sir. Thank you, sir.
Thank you.
Thank you, dear participants. We are starting with the session 2 that will be moderated by Dr. S. Saranya, Assistant Professor, Department of Biotechnology. Over to Dr. Saranya. Thank you.
Sir, நான் ஜாயின் பண்ணாருன்னா just So, Jayaseelan sir.
Yes, I'm available on the.
Available. Start பண்ணிக்கலாமா sir?
Yes, we can go ahead.
Okay, sir. Thank you, sir. Dr. Saranya will coordinate the session, sir.
Okay, wonderful. I think my background is blurring. Give me a second. I will switch off light and come back.
Okay, sir. Okay, sir.
So it's cloudy here, so I switched on my light, but anyhow, it's not working, it seems. Okay, of course, as most of you, I'm working from the home, sitting in the faculty towers of the university, but within the university, so more or less 24 hours job. Okay, yeah, please go ahead, sir.
Dr. Saranya will take over the session.
Okay.
Good morning, sir. Good morning to all the participants present virtually. Welcome back to the second session. On behalf of Dr. NGP Arts and Science College and ATAL FDP Organizing Committee, it is a great pleasure to introduce the guest of honor of this session, Dr. Jayaseelan Murugaiyan, Professor and Head, Department of Biological Sciences, SRM University, Andra Pradesh. Dr. Jayaseelan Murugaiyan obtained his undergraduation from Bardiyar University, post-graduation from University of Madras, Master of Philosophy from Bharathidasan University, and Doctor of Philosophy from University of Leipzig, Germany. Sir started his career as Assistant Professor in Biochemistry Department at Nandivarman College of Arts and Science, then in SRM Arts and Science College, Chennai, and Kongunadu Arts and Science College, Coimbatore. He then worked as teaching member at Belém Research School of Biomedical Sciences, Friedrich University at Berlin, Germany. Dr. Jayaseelan sir is an ad hoc journal reviewer of journals like Scientific Report, Applied and Environmental Microbiology, Frontiers in Microbiology, and reviewer in interdisciplinary science journals such as Journal of Pharmaceutics, the International Journal of Pure and Applied Analytical Chemistry, Environmental Pollution, BMC Research Notes, International Journal of Molecular Sciences, Journal of Integrative Omics, Veterinaria Italiana, and many other reputed peer journals. He is an external examiner of PhD thesis from several universities. Dr. Jayaseelan sir's area of interest are understanding host pathogen interaction using proteomics techniques such as two-dimensional gel electrophoresis, difference gel electrophoresis, and LCMS-based quantitative proteomics analysis. Then, identification of immunodominant proteins of infectious microorganisms using serological proteome analysis involving real-time samples, 2DE Western blotting, and protein identification. Sir is also interested in utilization of MALTI-TOF MS in species identification of microorganisms and higher organisms. Dr. Jayaseelan is a recipient of number of fellowships such as Indian Council of Medical Research, the Wellcome Trust, European Bioinformatics Institute, London, United Kingdom, and German Academic Exchange Service DAT Fellowship to attend a conference and deliver lecture at Shandong Agriculture University, China. Dr. Jayaseelan is a member of Protothecus Sclerosis Working Group. He is also a life member in Indian National Science Congress, Indian Society of Cell Biology, Society of Biological Chemists, Nutritional Society of India, European Peptide Society. Sir has around 41 publications in reputed journals and presented his research work in more than 56 national and international proceedings and conferences. With this brief note, I request our guest of honor to enlighten us with his knowledge. Sir, now the session is yours. Thank you.
Okay, thank you very much, Dr. Saranya, for giving an elaborative introduction about me. And at the first, I want to thank Dr. Sanmuga Raju, who is one of my best friends, for inviting me to this talk. And I also appreciate the participants from different universities and different institutions. I really welcome you, and I will start my lecture. So hope my voice is audible. Please tell me if you want to modulate anything. I will go with the slide now. Give me a second. So I have to go to these things. Right. Give me a second, so I will go into the things.
Yes, sir.
Are you able to see my slide?
No, sir. No.
No.
No,
Okay.
sir.
Okay. Give me a second, because many windows are open,
Okay,
and
sir.
we are used to. Okay. I will totally go with the screen, and then I will come back. Okay.
Okay, sir.
Give me a few seconds. No, it is. I can see the blocking, but it's not. I'm able to not to log in. So I think I have to open an other browser because the sharing light is not working here. Give
Okay,
me a
sir.
second. I will discontinue and then come back. Okay. I'll
Sure,
stop
sir.
presenting and then come back.
Sure, sir.
Sure,
Yes.
sir. Sir, now the slide is visible, sir. Yes, sir. You are audible, sir.
So now I will go into the sharing because there's some kind of things. Now you are able to hear my voice as well as you are able to see my slides. Is that right?
Yes, sir. Yes, sir.
Okay, perfect, perfect. Okay, so once again, good morning for everyone, and thank you for joining this session. And today I'll be talking about 2D gel electrophoresis, Western blotting, and protein identification as a tool for drug development. So before going actual into the topic, I just want to show you some introduction slides. So you see a life, it is beautiful. You see nature, again, it is beautiful. And if you look at this side, you have a tree of life. Everyone knows about the tree of life. We have a lot of microorganisms, lot of organisms, lot of macroorganisms. We know about this. And the other side, you see a life cycle of butterfly. Again, it's so beautiful. If you look at this butterfly, you will get a lot of questions. Sir, this is a textbook picture of, say, a 6th standard class. But what we are doing with the faculty development program? So the question comes, I have two questions. The first question, is the genome same for all the stages of butterfly? Say the egg, pupa, adult, whatever the case. Is the genome same? The answer is yes. When you talk about the proteome, it's completely different. Different structure, different shape. What about the brain? How it is acting? You look at the worm, this is an voracious feeder. That means it goes and eat the leaves. The legs has to be very strong, and the whole brain should activate only the legs and the mouth parts. But when it, from the cocoon stage, when it becomes the adult, a butterfly, the wings is flapping. That means the brain is also different. The brain has to activate the wing flapping, and the eating habit itself is completely changed. It's going to drink the honey. So this is what we are going to look. And with this basis, we will try to go what is proteomics and how this technology can be used for your drug discovery or for any sorts of work. Please, people who are listening to this, please feel free to ask the question. You can also stop me in middle, or you can also post the question at the end, whatever it is eligible for you or whatever is applicable for you. Okay, with this background, we again go into the basic unit. Again, this is the basic. The cell is the structural and functional unit of the cell. We know that this is a structural and basic and functional unit, but what is happening in the central dogma? We have genes that's converted to DNA, which we call as replication, and genes which is going to form an RNA, which called as transcription, and then protein. We know this, but what about the other things? There are so many things. There are intracellular regulations, the cell-cell communications, and so many things are happening. So this is what we are going to look. If you look at this basic slide, this was long back. We took this from an internet, from an article. And if you look at this, there are a lot of control. I told you the central dogma, the DNA, mRNA, and protein. You can see that a protein also gets degraded. It becomes activated or degraded, and it also undergoing post-translation modification. The mRNA gets inactivated or degraded. And if you look at this, there are a lot of control. All the blue highlighted boxes of the control, all these are proteins. You see that there is a control that decides the transcription. There is a control that decides the RNA processing. There is a control that decides the movement of the protein from the nucleus. And again, there is a control of translation. There is a control of post-translation regulations. There is a control of degradation of all the things. So it is all of protein. So many proteins are there. In this background, if you look at the proteomics, and we look at a little bit about the publications in 2003, exactly when Dr. Sarnia was telling that I was working in Kongunadu Arts and Science College. At that time, I was in Kongunadu. I was working with Dr. Sanmugraj also. At that time, there was a publication, Human Genome Project. So I was so excited at the time. I went to the classroom, the master's degree classroom. I told them, see, the human genome has been published in Nature. I can see the paper. And I was very proud to announce. And at that time, there was a lot of promises. You can cure any disease. You can have a designer baby. You can decide what sort of puppy cheeks or curly hair, whatever the things you want to do. That was the promise at the time. But after 6-7 years, in 2009, there was a Nature article. The title was Human Genetics Hit or Miss. That was the year where I did my PhD also. I did my PhD and finished. That during my lectureship, I was claiming, okay, you can do a lot of things. But at the end of my PhD, I was getting this question: how human genetics hit or miss? What is the question? Why this is one? We know the genome, but why we are not able to do the things? And in 2014, the first draft map of human proteome was published. It is only an 84% of the annotated protein of the human. That means this describes the known protein 85%. But what is unknown? There are a lot of things. So I will tell you this in next slide. But before that, I just want to introduce the terms omics. Everyone knows the study of gene is called as genomics, study of RNA is called as transcriptomics, and study of proteins is called as proteomics. And the proteomics, the term was coined by Mark Wilkins in 1985. He was a PhD student. Sorry, in 1995. He was a PhD student from Australia, and he was visiting Switzerland for a poster. Just for a poster session conference, and he need to write a term, the entire protein complement of the things. It's not possible, right? Because it's complicated. Because 10 times he has to write in the poster, and who will read that? And this is nasty. You can look at the same terminology 10 times. So he coined the term, what we call as proteomics, protein complement of the genome. So this was the birth of a new field in 1995. So okay. So look, what is this omics tells you. So here we introduce another thing called metabolomics, what we call the study of metabolites. So we have 4 different things: genomics, transcriptomics, proteomics, and metabolomics. And if you look at the other side, genomics will tell you what is possible. It will not give you exactly what is happening, but RNA will tell you what is probable. So that means it will tell you, yes, there is RNA is there, what is probable. But finally, the protein and metabolites will tell you what is actually happening. When you want to know what is actually happening in the cell, you need to do the proteomics and metabolites. And there's one more thing that is called as functional genomics. If you're studying about transcriptomics and proteomics, it's a functional genomics. So it will tell you what is the function. For example, in case of antimicrobial resistance, sometimes their gene may be present, but gene may be mutated. So my bacteria is susceptible to antibiotics. So Gene is present, it's also expressed, but it's susceptible. But we have to look that if the protein is intact, the protein is able to give you an answer. So that's what we are trying to do. So this is about the introduction, and with this background, we'll go a little further. We are and what is the difference? This is an important question. In the first slide itself, I told you the butterfly life cycle is completely a mystery. The early stage, the egg, the same DNA, again, the adult, pupa, worm, every stage, all 5 stages, same DNA, but the protein expression is different. That means the proteome is different. What you have in the first session, in the morning, and now you have, your proteome is different. In the afternoon, you will feel sleepy, you will feel hungry. All this proteome is playing a big role. So that's the thing. And again, the proteome is not a fixed feature of an organism. So you need to know the conditions on which the proteins is derived. So I have one question. Am I fast or my tone is okay?
Sir, no, sir, it is okay only. Perfectly
It's
all right,
okay.
sir.
Okay, very good. So then everything is perfect. Good. So it's the same genome, but different proteome. So this is the basis. And if you look at further, what is the complication? How complex is that? If you're studying about DNA or RNA, we have only 4 nucleotides, A, T, G, C, or U. So you can really know that. You can also sequence the 6 million base pairs of human being. It's very easy. The human genome is very, very simple. Now, if you pay 100 euros, you will get your genome sequencing. Some of the things, in my case, I used to go to some company and they tell I want the sequence. They simply tell that it's free for you. They simply do that. So that means it's become cheaper. But if you look at the protein, the complications arises. It's made up of 20 different amino acids. The first complication is that 20 different amino acids. On the other hand, if you look at the genome, I just take only the first statistical. It's not the exact number of genes. So you imagine this human gene has got 30,000 to 40,000 genes, and each gene can undergo 1 slicing or 3 slicing maximum, and we get 1 gene to 3 protein concept. So you will get 90,000 proteins if you take the lower level. So 90,000 proteins at any given time, and each protein can undergo 200 basic post-translation modifications. Imagine each one 200. At any given time, you will have 500,000 proteins in the cell. So are 5 lakh proteins in a given cell. And what you are going to do? Are you going to look at all this 500,000? Again, the complication is that. So is this the only challenge? No, there's further challenges. So I told you there are 500 cells, and we are talking only about a cell. And if you talk about tissue, how many proteins? Again, how many cells? Again, question. And go for organ. Multiple. Again, the organ, the proteins expressed in the kidney, proteins expressed in the heart, or muscle, or brain is different. So again, you need to read about that. So even the butterfly life cycle, you need to talk about the protein happening in the brain, protein happening in their body, or whatever the color. So everything is playing a big role. Then you go for the organisms. So organism level, the protein is very, very big. So we know our target. We can identify, okay, I want to go for single cell proteomics, or I want to go for an organelle proteomics, or I will take a tissue, say blood. Blood is a tissue. So blood is a, you know about it, it's a tissue. So you can take a blood, you can take urine sample, or you can take saliva, you can take CSF, or you can take anything. So you always decide. And if you are working with an organism, you will take an organism. So the whole organism. There are model organisms. For example, C. elegans. You can take a C. elegans, or Drosophila melanogaster, or zebrafish. You can take anything. So now you know a little bit target. But again, you know the complexity, the protein number may be very high. But is this the only challenge? So there are other challenges. There's a long-lived protein and short-lived protein. So the complexity comes with the long-lived and short-lived. For example, cyclins, which are involved in the cell cycle, they are just having the lifetime like this in 1 second or 2 seconds. So how you are going to catch them? The long-lived proteins is not a problem. You can isolate them, you can run, and you can get it. But the short-lived proteins, again, a question. And then there's a high abundant protein and low abundant protein. If you take albumin and globulins in your blood, they're present in tons. You can really do what are the things? I mean, tons means is gram or milligram. So you can really isolate them and you can do that. But low abundant protein, say hormones. If you take insulin hormone or any hormone in your body, it's in picograms. How we are going to study that? It's a big challenge. So the long-lived and short-lived is a problem because the short-lived proteins you cannot catch up, and long-lived proteins, it's a problem. I mean, you can do that. But high abundant and low abundant is a big problem because there are high abundant protein which can mask it, but low abundant protein will escape from your scanners. So that's again begin a challenge. And then again, labile modifications. The proteins undergo several modifications. You open it in the air, automatically all your methionine will get oxidized. Or So many other reactions. The oxygen comes in contact and then all methionine is oxidized. And because of the methionine oxidation, so many changes will happen. So this is labile modification. And there's no sequence facility, like mass spectrometry based sequence facility. It's a completely challenged because it's not established yet. And again, the protein arrays and antibody arrays, we call it as microarray simply. This microarray is not complete. Again, it's not simply it's not possible. And there's no PCR-like technology to get the multiple copy of protein. I'm talking about hundreds of protein and thousands of protein. You may recollect Merrifield's solid phase protein peptide synthesis. That's a very challenging one. That's applicable for only one protein of medical importance. But again, it is not possible for us to go for investigation level. It's a big problem. Is that the only challenge? Again, the challenge comes. Say, the cellular localization, where is the protein present? Is it in nucleus or mitochondria, endoplasmic reticulum, Golgi apparatus? So you will have lots and tons of questions. Where is it exactly located? For example, HSP 70 or 90, HSP 90, heat shock protein 90. If it is present in cytoplasm of a microorganism, it's common. Well, the microorganism is quite uncommon. But if it is present on the surface of the microorganism, this microorganism is infectious in nature. That means that can cause disease. Same, a single protein which is inside, okay, but it's when it's outside, it's causing a disease. So you need to know what is the exact location and function. For example, you have multiple isoenzymes. So what is the precise function? How they coordinate with themselves and the pathways involved? Thousands, millions of pathways. How we are going to do that? But these are all big questions which we need to answer. But at the same time, now the real question comes, how we are going to go for thousands of proteins at any given point. So the technology was developed in 1975. So this 1975, they started the technology what we call as two-dimensional gel electrophoresis. I'm not going to talk about STS page, but I have slides for you to show if anyone has a question. But the two-dimensional gel electrophoresis, it applies in both the ways. We separate the proteins on their PA value, that are the isoelectric points. So for example, on the top, you can see that first we are separating the proteins on the PA value. So PA is 3 and 200, and then we are separating the proteins according to their molecular weight in the case of second dimension. This is a silver stained cell of a human T cells, and this was my own cells. So most of the pictures are my own thing. So this was the first one we isolated, and we were able to see a lot of protein. In fact, there are 3,000 proteins visible in this gel. I tell you at the end of the presentation, or the mid of the presentation, how we are going to quantify them. That's what you will not hear from other classes. Since it's an FDP program, that's very important, where you can go for quantification. Everyone knows what is 2D. You can also know theoretically or practically. Some of them might have done the practical things, but I'm going to tell you what is done, can be done with the things. Okay, so the two-dimensional gel electrophoresis is one of the techniques which gives us the answer, where you can go for separation of hundreds or thousands of the protein. So with this background, what is the workflow? We go into the proteomics workflow. It's very, very simple. It's composed of 3 different stages or 3 steps. The first step is that you can see that step is also available. The first step is that sample preparation. The second step is the sample separation or protein separation. And third step is the protein identification. So we are going to dwell upon only these things today. So the first one is the cell lysis and protein solubilization. So the important thing is that you need to break the cell open and bring the protein into the solution. That's the only task here. You can use any method. You can use, say, physical method like pestle motor, or you can use a rotor, you can use a ultrasonication for instance. So the physical method is that. The chemical method, you can use some chemicals, osmotic imbalance, and you can cause the imbibition or crenation of the proteins, the cells, and we can open the cells. So one is physical. Now we have done chemical. The third one is the enzymatic method. You can use the enzymes to break open the cell and bring the proteins in the solution. So the decision of your research starts with the protein solubilization and lysis. How we are going to solubilize, how we are going to lysis, and with minimal impact. You can see that there are a lot of proteases enzyme within the cell. When the moment you open, these proteases will act upon the other protein, and thereby it can damage the other proteins. So you will not get an exact picture. So you need to have a protease inhibitor of the sample. Or I use in the laboratory a flash freeze. That means I will stop the reaction by either directly taking it to the liquid nitrogen or denaturing it very fast. I use acetonitrile and the ethanol to break open the cell, to digest the lipids on the membrane, and acetonitrile to bring the proteins into the solution, or denaturing it. So once you denature, you will get it. The second concept in proteomics is that protein separation. This is very, very important. Please pay attention to this. There are 2 different technology. One is gel-based technology. Other one is non-gel-based approach. On the top, you can see that the gel-based approach, we use STS page. polyacrylamide gel electrophoresis, or 2D gel, in two-dimensional gel electrophoresis, or DIGE, differential gel electrophoresis. And then once we get this gel separated, we go for cutting the spot or band on the gel. We go for triptych digestion. It's trypsin treatment. Technically, we call this a triptych digestion. Okay, so make sure that the triptych means it's trypsin digestion. So it's a technical term. So we go for triptych digestion, and then finally we go into the mass spectrometry for base identification, and we search in the database. We get a protein list, and we go for the bioinformatics. This is one workflow. This is called as gel-based approach. So you are using a gel. Then on the other hand, we have a non-gel-based approach. We use several technology. For example, isotope-coded affinity tagging. This was my first technology. It was very expensive, but I like the technology. But you can also use the label-free quantification or direct mass spec. What we do is that we do several things. We separate them using HPLC, high pressure liquid chromatography. It's otherwise called as high performance liquid chromatography. So again, there you can use that, and we use at a nano level because we have issues. Our sample is very less, and then we can go take this to the mass spectrometry, and the rest of the work is go. So again, I repeat this. So the important concept is that only three: the sample preparation, protein separation, and protein identification. In the first one, this the sample preparation, you need to break the cells open. This can be achieved by physical, chemical, and biochemical methods. These are the three methods. And then this protein separation, this can be achieved by two different steps. One is called as the gel-based approach, and the other one is the non-gel-based approach. Both the approaches, we go for the protein identification using mass spectrometry, and then we go for the database search, and we go for the protein list, and we go for the bioinformatics of differential and expression analysis. So it's mostly the bioinformatics most of you might be using. In the gel-based approach, we have SGS page, we have 2D gel, we have the differential gel electrophoresis, and Western blot. So today in this session, I'm going to deal only this aspect: gel electrophoresis, DIGE, and Western blot, and we are going for the mass spectrometry, how the identification is done. Okay, with this background, we'll see how this technology can be used for the drug and protein expression. For example, you have an effect of a drug on protein expression of a particular cell. It can be done in vitro, in vivo, and in situ. That means in vitro, you can take a cell line and you can do it in the laboratory. You can expose to the drug and you can see what happens to this drug, or what's the influence of the protein expression. Or you can also do the in vivo or in situ. You can try to do any of these things. Whatever the technology you use, I told you isolation of the protein or extraction of the protein. You just go for the methods. You can isolate the cells, or you can rupture the cells and get what you call as a protein extract. Once you get a protein extract at protein level things, you will go for different technologies. The first technology is the biochemical analysis. You look for, it's highly specific. It can be an enzyme, it can be a protein, or it can be coupled with something else. So you can do whatever you want. It is a single proteomic approach or single parametric approach. You will look for one particular enzyme or one protein or one hormone, something like that. You can look only one at any given time. It's something like this. You are going to a pond with a fish and bait. So that means you will have only one stick and you are catching only one fish. That's the biochemical. It's highly specific. Okay, it will definitely fetch you a fish. Next is the proteomics analysis. I told you what is proteomics. It's a global level and it's a whole proteome. And then here again we have gel-based and non-gel-based. The proteomic approach is that you are having a fishing net. You're going to a pond with a big fishing net, throw the fishing net and get all the fishes. So it's a complete proteome. And there you can get both upregulated proteins and downregulated proteins. So that's what you're going to do. But in case of biochemical analysis, it's only one. But in case of proteomic approach, it's complete. So you're going to catch up all the proteins from the cell. So see, it's not all 500,000 proteins, because I told you there's a small amount of protein or low abundant protein and high abundant protein. So the high abundant protein is something which you get it. For example, you are taking a blood sample, you will get albumin and globulin, and you go for depletion kits. The depletion kits means you are going to remove the albumin and globulin and you're going to get the low abundant protein. For example, I can tell you an article. It was published in 1970s, I believe so. There was one professor, that is, I mean, he's still live in Italy, Professor Rigatti. He wrote an article called as the democratization of proteins. What is the democratization of proteins? That's a very good question. If you lift all the big proteins from the sample, say serum, then you will get the smaller proteins visible. So you are lifting all the one which is dominant over, and so the smaller proteins will be visible. That's what we call as proteomics technology. Then we have a microarray technology. It's highly specific again, and the technologically limited, because you need to print these proteins on the microtetra plate or on a plate. So that's a big thing. So microarray, if you want to talk, we have to talk another 6-7 hours. But it's anyhow, this again specific. The technology is not developed. So the center point of the technology is the proteomics. So now with this background, we'll see how this 2D is done, how this batch is done, and how we are doing Western blot, and how we are going into the protein identification. So do you have any questions until now? Anyone can answer. Any questions? I don't hear anything.
Sir,
Did
no,
I ask
sir.
some question? Did I ask some question? Hello?
Sir, no, sir, no.
Okay, so my speed is okay, right?
Yes, sir, you are
Okay, very good. So then we come across the two-dimensional gel electrophoresis. Some of you might have done that. It's very simple technology. You have a strips. Here in the second picture, IPG strips, you can see that there's a faint colored one. You can see the strip. This is a gutter where you put your sample. This is the strip. This is a commercial strip. So in fact, it has a one positive end and negative end. Different PIs is fit here. That means different PIs 3 to 10. Okay, there's a broad range. I will tell you in next slides how is the broad range and sort of. So the buffers are kept fixed on the things. So we have a PA strips, and then what we do is that we first go for the isotope focusing. IPG4 is a machine. This is from GE Healthcare. There are Biorad, there's Sigma. So many companies are also producing. My favorite machine is GE Healthcare. That's why I sold my own machine. So this IPG strip machine is there. So once you go, you take the sample and you put them on the cutter, and then you keep the gel on the top. So this small gel, this IPG strip will get will uptake all the samples. Once it's uptaken, then you fit it here. You go for isotope focusing. Once the isotope focusing is done, what you do is that you're going to add. I'm sorry, this meter is a gas meter. This neglect it. This is a 24 centimeter big gel. And you can see that the strip is layered over the top of SGS page. Already we have separated the proteins based on their PI, and now we have layered the proteins, this thing over the top of a SGS page, and then we seal it. Once it is sealed, then we run the second dimension, and then we get what we call as the silver stain gel. I mean, it's only an example for silver stain. I will tell you the staining later. But this is one of the example which we get it. So you can see that PI, and then this is one. They may be differentiated based on the molecular weight. So this is about 2D, and then again the sample preparation is very important step. I told you need to break all the macromolecular interactions. So all disulfide bonds has to be removed, and then all the substances which you believe that it will interfere with your focusing, isotope focusing, say salts, polar detergents, lipids, polysaccharides, nucleic acid has to be removed. So normally we remove in a very good way, because we go for liquid extraction and then we remove that and keep proteins in the solution during SGS page. So the important concept here is that the carry home point is that all the proteins should be in the solution. That's what we want to do. And again, there's no universal solubilization protocol. Every lab uses, most of the lab uses their own technology, and they use urea. So we use urea, and then this urea will keep the protein in the solution. And then the contamination removal, I told you here, the salts, polysics. You can do dialysis, you can do filtration, centrifugation, chromatography, and solvent extraction, which is called as liquid extraction. You can use any of this technology get rid of the impurities. Normally I use a centrifugation. I just rupture the cell, I centrifuge, all the precipitate will go. The DNA, impurities, cell organelles, everything will precipitate. I will take the supernatant, clear supernatant, and this supernatant contains the proteins and will proceed towards the things. And then there's a thing called a sample preparation. This is very important. So you need to have a 9 molar urea and plus 7 molar urea. Whatever you calculate, it comes 9 molar. Ninth thing, you can see that either 9 molar urea and 2 plus 7 molar urea. Anyone can tell me, if you have time, why this 9 molar? Can't we make it round 10? Okay, I don't hear anything, but anyhow, the 9 molar urea is the saturated level. If you add 1 salt more, it will not dissolve. So this is the thing. I think someone is texting a message, but I cannot see the message because you are able to see my slides, right? Excuse me, are you able to see my slides?
Sir, yes, sir.
Yes, sir. Yes.
Yes,
Okay,
sir.
the problem is I'm used for Zoom and I never use this Google Meet, and
Oh.
I don't see anything in the side.
Okay, sir. She answered as pH. You asked the question, one person answered
No.
as pH.
No, it's not pH, it's saturation. The 9 molar is the maximum molarity you can prepare. Or if you can use 2 molar thiourea and 7 molar urea. Why so? Because the urea can make changes on the amino acids. So that's why we try to reduce the concentration by 2 molar, so that goes in thiourea. And we need to have a surfactants. There's a chaps and other things. You will use it, and you will also have a reducing agent, which is basically a carbohydrate molecule, either DTE or DTT, and then you use an ampholyte because you need to maintain the pH of the strip. So this is what we normally use it, and this is the tip. If you know this, if you master over this particular process, then you are the boss of this particular field. Okay, we go to the next thing, the immobilization. How we do that? So I told you there is a strip. The strip can be pH 3 to 10. This is the strip. It's a commercial one. It's 10 euros per strip. So approximately, say 800 rupees per strip, but it's cheaper. When you compare to, when you compare to preparing yourself in the lab, it's very, very cheaper. And after all, it's 12 strips per 100 euros. That means approximately it will be around 8,000 rupees. And you can get most result. I'm talking about 24 centimeter strips. Then what we do is that we need to dehydrate. The strip comes as a dried one. It's completely dried one. What we do is that we prepare the sample from the previous thing. So from the previous cell, you can prepare the sample with mixing all these things. That's the protocol to mix it. You will see the practical approach. You mix the sample, and then you add the thing, and then you put them on a gutter, and then put the strip overnight. It's a passive diffusion. The samples will get into the strips. So once you get into the strips are ready. We call this ready for the IPG, ISO ultra focusing. So you can do it the next day. There are commercial strips, so something like this. You have 24 centimeters, 18 centimeters, 11 centimeters, and 7 centimeters, all from different companies. You can go for Biorad, you can go for GE Healthcare, you can go for Sigma, or any of the local companies. You test it. When you want to start the 2D, you please test it. Get all the strips and then to do the testing, and you can do it. And you can go for the thickness. Thickness is uniform across all the company. It's 3 mm wide and 0.5 mm thickness. This 0.5 mm, you can also see them. Okay, you can really see them on your naked eyes. And then there's a glass plate. On the glass plate, this gel is fixed with a different pH. Okay, again there is a broad range and narrow range. For example, there's a mistake here. The broad range is 3 to 10. Narrow range is 4 to 7. Maybe you will correct it. Otherwise, say example 4 to 7. So the broad range is something very big, and there's an ultra narrow range also. For example, you want the pH between 4 to 5 or 4 to 4.5. So it's very clear. So you will get all the proteins which is 4 to 4.5. So that's what you will get in those strips. So again, there are commercial strips. It's from GE Healthcare or Biorad or Sigma. You can choose from another local company also. And the pH range, there are broad range strips because it covers from 3 to 10 or 3 to 11. 3 to 11 is sufficient. I don't use 3 to 11 for some reason. And there's a gradient gel and linear gel. The linear gel means you will have a gel piece and all the regions are equally divided for even individual pH. So for example, 3, 4, 5, 6, 7, 8, 9, 10, 11. It goes like that. Or maybe 3 may be so big, 4 is little big, and it goes like that relatively. We call it as an it's a gradient system. So it's something like that. So you can go for linear or non-linear. So the linear means it is divided equally. Each pH is divided equally. It's not in the slide. You may note down. Non-linear means each pH has its own range or the length. Okay, so what do you do in the first dimension? You have already focused yourself. You have already made the sample to enter into the gel. So you have the first dimension strip. For example, 7 centimeter strip. I take the example, and 3 to 10 NL, the non-linear range. And I dehydrate with thiourea, chaps, and DTT, ammonia citrate, and iodostamate. So this is what I use it. The urea thiourea 9+1 formula, and then I do it passive dehydration. After that, I go for iso focusing in the machine which I showed you earlier. It will take 6 hours 30 minutes. That means the focusing will have different steps. The step and hold is something like this. You have 300 volts, and this 300 volt is maintained for 1 hour continuously. But there is a gradient volt from 300. It reaches 1000 in a range of 2 hours. So I decide. Okay, it goes like that. So that at the end of 1 hour, it will be 301, 302, 303, 304 like that. It will reach 1000 volts. It's gradually increasing. The voltage is gradually increasing, and this happens in the isotope focusing for 2 hours. And after that, again, there is a gradient. It will reach 5,000 volts. And again, it go for 2 hours, and the last step is 1 hour 30 minutes. The step and hold, we apply 5,000 and maintain it. At the end, 6 hours 30 minutes is the running time. Then you imagine on Monday you rehydrated, and Tuesday 6 hours 30 minutes focusing you have done, and you can take them in the freezer, or you can proceed for the second dimension. So what you do is that you use a treat with DTT, break the disulfide bonds, iodostimate to stabilize it. So this is very simple concept. DT to break the disulfide bonds and iodostimate to stabilize the other things. And then you go for the second dimension. This is a normal SGS page. And if this is a small gel, you apply 10 milliamperes, 1.5 hours. If it's a bigger gel, 24 hours. I'll put 24 hours running. That means I will start today morning at 10 o'clock. I'll finish it the next day morning, 10 o'clock. I'll put 1 watt per gel. If I have 12 gels, I will put 12 watt per gel, and then it's a circulating unit. I just leave it, and this will go for the second dimension. So before going in the separating and staining, I want to show you what happens to your protein. So if you have a protein which is PA5, the isotope focusing, the IPG, it happens precisely. If your protein is here, it automatically, so your protein PA is 5. It's less than this, the PA is 3, so it has got a positive charge. That means it will move towards negative charge. At the same time, if the protein is here, the PA has pH of 7, the same protein will have a negative charge. So it will move towards a positive charge. Once again, repeat, the protein of PA is 5, and if it is in this region, not other than the PI. The PI means is the isotopic point. The isotopic point means it is the pH at which the protein's net charge is 0. So net charge of the protein is 0. Even if you apply a lot of electricity, they will not move because they're not charged. Only the charged particles move in the isotope focusing. So that's the concept we are using it. And what happens is that, say the protein is here, PA5, and if the protein is here, it will have a positive charge and it will move towards this side. And if the protein is here, it will have a negative charge, it will move towards the positive side. But whatever the case, whether it's moving this way or this way, they will come to the PA5, it will lose all complete charges. Complete charges is gone. And then it will not move further. That means you are separating the proteins based on their PI. Each protein has their own sequence. Each protein has their own PI. So you are separating them in the things. And again, we are going to separate them in SGS page. So in SGS page, stacking gel and separating gel. For 2D, we don't want any stacking gel. I'm not going to answer this. Why? What's the stacking gel? You are already professors. So this this stacking, instead of stacking, we use the IPG strip on the top and seal it with agarose, and then we run the things. So this is the same thing I got from the internet source, and you can see that it's from a book source. So you separate them and separate the proteins. Once you separate them, you're going to stain them. I'm not going to go into the protocol, but I'm going to tell you what's this different types of separation. We use Kumasi, R250 and G250. G250 is mostly for your protein determination by Bradford method, but R250 is for staining the gels. And we prepare colloidal Kumasi, and you can see the sensitivity is coming very, very good. So 5 to 10 nanogram per spot. So some single spot can give 10 to 5. And we have a silver staining, which is 1 to 4 nanogram. So if the spot is 1 nanogram, you can see that. And we have a copper stain, zinc stain, which we normally don't use it. And then we have cypro ruby stain, which is a more sensitive, it's 1 to 10. The silver staining seems to be the better, but again, the silver staining will cause environmental problem. So I don't use silver staining. Please note down, we use a blue silver staining in the laboratory. Please, you can refer it. It was published in 2004 from an Italian group, and we use the formula, or we use the technology to use stain our own gels. It is called as blue silver Kumasi staining. The Kumasi, if you look at that, see, it has an interesting story. Do you know where we use Kumasi in our day-to-day life? Anyone can answer. If it is chatted, please, the moderator can highlight to me. Do you know where we are using Kumasi? Yes? It's a question. Mostly Tamil, no? You know that the blue sotanilam. You know that in order to maintain the whiteness of the dress, we use blue drops. Yes or no?
Yes, sir.
That's a Kumasi. And some of the cakes, they appear blue. Cake, we use in cake. Okay, sir, you're telling the laboratory Kumasi, can we eat it? No, this is chemically synthesized. Be careful. I tell you, in Africa, we have a region called Ashanti in Ghana. In Africa, there's a country called as Ghana. In the country Ghana, there's a region called, like Coimbatore, they have Ashanti. And Ashanti region's capital is called as Kumasi. So Africa, Ghana, and Ghana's one of the region is Ashanti. Nice Ashanti, like Ashanti. Then the capital of Ashanti is Kumasi. So when the British started exploring those regions for making themselves slave or whatever it is, they saw that these aborigines or the native people, they were painted with the blue color, and the hot sun, it was glowing. So they thought it's called as Kumasi because in the name of the name of the city. At the same time, there was one German scientist who was able to synthesize this chemical Kumasi, and these people carried this to the England for textile dyeing. So they need to use this for the dyeing industry. So they need to have a colored cloth. So they used this Kumasi. At the same time, the hospitals on those days, when the textile workers used to go to hospital, they will go with a blue stain on their body because they don't use gloves on those days. And then the doctors were worrying, what is this? Hey, what is this blue color? They said it's a Kumasi and it's used for staining the dress. And these doctors started using to stain the proteins. That's how we got it. So this is natural. You can pour them in the sink, it will get degraded. So I strongly recommend you should use Kumasi, not silver staining. Silver staining, you know that it will cause an environmental problem. You know much better than me what happens to the environment. It goes to the fish aquatic system, and then it comes back to your table. It will come to your dining table. That's the problem. Then you consume it. So the silver, whichever you throw in the environment, never ever escape from the silver you throw into the environment. It will come back to you. So be careful, and we use Kumasi. I strongly recommend blue silver Kumasi staining. Okay, again, it's a limit.
So can I ask you a question?
Yes, go ahead.
Sir, in the protein, where is the binding site of the chromatin?
Oh, there's tyrosine, tryptophan, and phenylalanine. So there's a big bonds that goes into this site. Okay, tyrosine, tryptophan, phenylalanine. And there are other amino acids also. It can go for hydrophobic interactions, and it's the same thing. I tell you, I asked you a question. This is a very good question. When you see a gel, SDS-based gel, you see on the top, you can see that very intense bands, and in the down, you see a very light band. You know why? Because on the top, the protein is bigger. There are a lot of phenylalanine, tyrosine, tryptophan. It goes and binds. And once the one Kumasi molecule binds, the second molecule will go and bind to this. So we have a stacking effect. That's why you get a much more intensive band on the top, and in the low molecular weight region, you get less intensive bind. Is that clear?
Yes, sir, sure. Thank you very much.
Very good. Okay, so I want to go back a little bit. I want to ask you one question and then I go things. So here, this one, you can see that I put the gel, I go to the molecular weight in Dalton, in kilodalton. I started with 210 and I go with 10%. So that means there should be some drawback. What is the drawback? The first drawback is the molecular weight. Those protein which are more than 210 kilodalton, say membrane protein, you know about the G protein, transmembrane proteins. So all those proteins, you have this 210 and more. If your protein is 250 dalton, it will not enter to the gel. I'm talking about 12% gel. There are different types of percentage of gel. That if I want to take only STS page, it will take 3 hours for me to complete. But again, I tell you, this 12% gel cannot permit 250 kilodalton protein. That means all those protein which are bigger is completely lost. So this is first drawback. The second drawback, I make it very clear. This bromophenol blue dye is here. It is only 10. That means those protein which are more than 10, less than 10 kilodalton. Now I'm talking about the protein which is less than 10 kilodalton is lost. It's already ran over the gel. So the first drawback of two-dimensional gel electrophoresis is that the proteins which are having higher molecular weight, say 250 kilodalton, is lost. First. The second one, the proteins which are less molecular weight, less than 10 kilodalton, is lost. You can go for peptide gel electrophoresis, again, you will lose some protein. So the proteins of higher molecular weight and lower molecular weight is lost. And then what about the hydrophobic proteins? The proteins which hate water, they will not mix it. They are also lost. You see, Now we have got three drawbacks. One is the proteins which are higher molecular weight, more than 250 kilodalton is lost. Less than 10 kilodalton is lost. Third one, hydrophobic protein is lost. And those who have got an extreme pH. So we are using only pH between 3 to 10. What about 11? What about 12? What about 2? What about 1? They're all lost. So the proteins of abnormal pH or an extensive pH is lost. So three drawbacks we have seen. The proteins which are higher molecular weight and lower molecular weight will be lost. Then the proteins which are hydrophobic in nature is lost. And third one, the proteins which has an abnormal pH is lost. So if you look at this thing, those are the drawbacks. Okay, those are the drawbacks and which I have given you. And one of the drawback of the limitation is the lack of resolution. So you have seen these spots. Some are faint, some are good, some are fused, some are not true things. It's a big issue. And again, if you look at this reproducibility of the gel, I run the gel in my lab or laboratory in the morning 10 o'clock, and I'm using the same sample, same gel, and same apparatus, everything. And I'm running the gel tomorrow morning at 10 o'clock. I will get gel to gel difference. Definitely, I will get a gel to gel difference. What about you, sir? You come to my lab, you work in my lab, you will also get a different gel. So that means a gel to gel differences between, I'm talking about within the laboratory. So I go to your laboratory and my laboratory. In your laboratory, the same sample, same solutions, you will get a different gel pattern and I will get a different gel pattern. So it's gel to gel, running differences will be always there. So how we are going to do that things? So in order to do that things, we use a differential gel electrophoresis. In other words, it is called as multiplexing. So 2D differential gel electrophoresis. This is a wonderful technology. It was quite expensive. Now it is very cheap. And what we do is that, imagine, pay attention to my words now. So we have sample 1 and sample 2. Sample 1 is 150 microgram of protein. Sample 2 is also 150 microgram of protein. What we do is that from sample 1, we are taking 50 microgram, adding into the center of our, we are making it 1. And from here, you are taking the sample 2 also 50 microgram and putting it here. At the end, now you see, we have sample 1, sample 2, and a mixture of sample 1 and sample 2. And if you look at the amount of the protein, it is 100 microgram of sample 1, 100 microgram of sample 2, and 100 microgram of sample 1 plus sample 2. That means you will have equal concentration or amount of protein in all three. So this center one, the pooled one, is an internal standard. That means you have 100 samples, or say 100 is too much calculation for you. Maybe we can take 5 samples. So for 5 samples, you will get 6th tube. So for all the tubes, you will take equal proportion, and then you will reach the 6th tube as equal one. So that means you will have an internal standard. What we do is that we label with a side probe. I mean, side dice. We have side dice. I don't need to explain what is side dice. So we have side 2, side 5, and side 3. There are 3 types of dice. Of course, there are other dice also, but these are the 3 predominant dice we use it. And what we do is that we label the pooled sample with side 2, always side 2. Keep in mind, always side 2. That's the internal standard. Then side 1 goes to the side 3 goes to the sample 1, and side 5 goes to the sample 2. And what happens is that you are mixing them together because it's already labeled. The sample 1 is with side 3, sample 2 is with side 5, the pooled sample is side 2. You are mixing them and running as a single gel. That means you are running 3 gels at any given time in a single gel. And then you will get the image like this. So you need a differential gel scanners, and the scanner will get an image like this. So you will get a green color for sample 1, and red color for sample 2, and blue color for this one, this sample pooled one. So you get a 3 image, and this is the fused image, which will get a golden color. You go further. So this is what you really see that. If you look at this gel image, these are Jurgat T cells. The T cell lines we expose to a benzoazopyrin. So we expose to the benzoazopyrin, and we do that. In this slide, what you see is that these T cells were stimulated. You know about the stimulation in immunology. The PMA, propanol mestic acid, and ionomycin, you add it and you stimulate them. So what we do is that we have an unstimulated cell and we have a stimulated cell. These 2 samples are treated with 2 different dyes, and the internal standard also you're calculating, and you're getting a gel image like this. Whichever the protein is higher expressed, you will get signal. For example, this one is green. That means the sample 1 it's higher in unstimulated cells. Whenever you get a red, it's the sample 2. That means this stimulated is 2. That means you can also know that up and down regulated proteins. And you are also minimizing the gel to gel variation because you have an internal standard which is common for, say, you are running at 12 gels. So all 12 gels will have a same internal standard. So what you can do is that you can take the internal standard, you can calculate everything and get it. So this is what the differential gel electrophoresis. There is a lot of technique behind, and in order to do the quantification of this 2D electrophoresis, you need to scan it. So how will you scan it? I stop here and I want to ask a question to the audience, especially. How will you scan a gel? You have a special scanner from GE Healthcare. It costs 8,000 euros, and I don't want to purchase that. Why should I purchase 8,000 euros a scanner? How can I do the scanning? It's open for anyone. I did not hear the voice. The voice is echoing. How will you scan a gel? Such a simple, huh? I tell you the secret, man. Don't worry. You will use an ordinary paper scanner. Say Liddy scanner, which is how much it costs? 2,200 rupees? Less than that. I'm pretty sure less than that. It's a 60 euros equipment. We just put the gel on the 60 euros equipment. 60 euros means it should be approximately 2,500 or something like that. Not even 2,500. Less than that. That scanner, we can get it and keep it in the laboratory. You can scan it at 600 dpi. 600 dpi means for the publication you need 300 dpi. So scan it at higher level. So in our laboratory we always do at little bit higher level so that it will maintain the standard that the needed things. So use a scanner and you get it the things. Only thing is that you need to know how to scan it. So once you scan the things, you get a good image and then you go for the gel analysis. There are several softwares to do the gel analysis. This is the point which is important for you because the 2D gel electrophoresis, you know where it is and how to do that, and you know the equipment. But here, this is the challenge. What you need to do that. There are a set of softwares. Give me a second. There are a set of softwares. You can use Delta 2D software. You can use a Decider from GE Healthcare. You can use the Image Master or PDQuest or Prognosis. But in my opinion, the Delta 2D works fine because I was working for more than 15 years with Delta 2D. Now, yeah, 15 years. It's very good. Decider, I worked it. The Delta 2D is something like 10,000 euros. You can also bargain it. You can get it down 7 lakhs approximately, 7 to 8 lakhs. It's an image processing software. The Decider is something like 25,000 euros. It's much expensive and the service is very poor. Even though GE Healthcare, now it is CTV is the name of the company. But this one is, I don't recommend it because I purchased that equipment and I never used it. So, and Image Master is also good, and PDQuest is also good, and Prognosis is also good. And if you go for the Prognosis, PDQuest, and Image Master Delta 2D, you can ask for a student version. That means, sir, my student has a gel. I want to do the gel for, say, 1 month. I don't want to purchase it. I don't have money for that. I don't want to spend 7 lakh rupees, but they will give you a 1 month license for free. That's what I want to tell you. So you can use it. Okay, we'll see with this thing. You need to have certain procedure to scald the gel. So the stain the gel optimum. So don't stain it very intense and very low. Stain it at optimum and no gel breaking. Most of the times we will face gel breaking. Gel will break like anything. So avoid gel breaking and avoid background artifacts. And noise. Noise is the dust. So keep your lab tidy, and then you will get anything. And most of the times you process your gel, stain your gel under the bench. Under the bench means laminar airflow or laminar air hold. So don't switch on the fan, but you keep them under the bench. So the most of the things, most of the impurities and dust will not come inside. And moreover, you people will are getting keratin, including me. You will get keratin falling down from our skin, even though you're covered. And some people will come with a loose hair and then do like this. So don't do that. So keep them in the bench. So something will take. And you should also avoid insufficient staining, contamination by fingerprints. You should not touch it as you like with it. Use gloves and fluorescent sprinklers. Sometimes you use fluorescent sprinklers and bubbles that will create a problem. So use a grayscale. Normally, I could scan the gel in 2 ways. One is the grayscale, other one is the color images. Because this is very important. The color image goes for the publication, grayscale goes for the analysis. And then you can also limit the post-processing. Exactly the gel image, exactly the position. You don't do extra things and you can cut it at the end or whatever it is, and no mirror image. So keep the gel in same position. If you take 12 gels and at the end, if your professor or you yourself happen to see the 12 gels, you should not see the difference. Oh, everything is taken in the same yard scale. That's why you use a little paper scanner, and there you can see that A4, A3 markings, and you can use these markings to guide yourself to fix that thing. And you also pay attention to the file format. Avoid JPEG. Most of the people are happy to go for JPEG. Don't do that. Go for TIFF. TIFF, LZW compressed. That means that's the easy trick to get it. Don't go for the image analysis. So once image analysis, you need to manipulate, you need to spot detection, and you need to go for the comparison and data analysis and data representation. So you have 10 gels. First 10 gels, I want to go do the comparison. What I do is that I layer one after the other. What we call as the overlapping gels. Then you normalize. Then you create a master gel image, and then from there you spot the detect. I mean, you name everything, and then you calculate it, and you compare the gels, and finally go for the data analysis. Maybe I can explain you with the Delta 2 software. What we do is that we create a file, importing of the gel images into the software. That's the first one, and you can also see the 2D image or 3D image of the view of the spots. That's what doing. You organize the gels in such a way, and the step 2 is that warping. Keeping one after the other. So physically you keep one after the other. That's the thing. Then you fuse it and make a artificial master gel. Once you warp it, you have 10 gels or 12 gels or even 24 gels. All these gels you warp it, and then you get one single image, what we call as master gel image, and you detect the spots on the master gel image. And if there's one, sometimes there will be 2 spots will be fused, sometimes it will be marked as 1. So you make them, tell the software, no, it's 2 spots, you go like this. So you guide this software, then you label all the things. You say 2,000 spots or 10,000 spots, you label all of them, and then you go for transfer to the gels, and then you get it. So this is what the reference gel, and you're combining and creating a master gel, what we call a typical profile. Then you can transferring this into the individual gels. At the end, you will get something like this. So you will get an information, something for every spot. I'm talking about every spot. There's a statistical thing, and you can see the 3D view. You can turn, you can see whether it's a view or some speckle or some dots or whatever it is. So you can get it. Then you will get what you term as this up and down regulation. So then you will express these things. Okay. You know that you can do that analysis, and then you want to upload it in the database. Because if you don't upload in any database, 2D gel databases, your gel, who knows that you have done a gel or not? So you need to fix the gel. You need to dry the gel first case, keep it safe in your lab note. And the second thing is that you upload in the gel data bank. Gel data bank, there are several data banks, and what I want to tell you is that Swiss 2D gel page, we have a gel bank, and we have 2D page and world 2D page. So if you go to the world 2D page, it's the Swiss bank, it gives like this. Because of the time constraint, I don't want to give an online demo. I thought it will go faster, but I'm going very slow. So you can do that. Maybe tomorrow also I have a presentation. At the time you can ask me. You go into the things, and you can browse it. You can find out what is what, and you can come back. So you need to export and upload into a database. So you're going to you upload into the 2D database, Swiss 2D database, and then it comes. Or if you don't like that, you can go for the gel bank. This is how it looks the software. This is the link here given, and this one you can do it. Why should you upload a gel in a data bank? If you want to publish it, most of the authors, most of the reviewers, most of the journals, they like first, okay, where's the original gel image? You should not manipulate, right? There should be an evidence. So it's called as 2D gel mapping. So they will ask you, where is it? So you can add that. It's free. Uploading is free. You're not going to spend anything except your time and energy. So once you have this, so then this will go. And again, this is the page for the third page for 2D, world 2D page, where you can add things. Okay. So do you have any questions in 2D so that we can go into blotting? Or you can also ask me later also, no problem.
Seems, can you show that that picture in which you have shown the green and red diagrams, like upregulated and downregulation?
Yeah.
Yes, sir. Yes, sir. This one. Sir, how we can say that this is the upregulated and this is the downregulated?
Okay, that's a good question. What we do is that this is a fused image. We call it an overlay. 1, 2, 3. The gel scanner will scan 3 times. Okay. 3 times, and then this is a fused image. If it's golden color, that means these two colors are equal proportion. It's green and red. If this is less concentration, green is less concentration, you will get red, more intense. You can see here. This green is more, you can see that this one. So in this case of red, the green is less. In case of green, the red is less. That means the concentration of this particular spot, a particular protein is more or less. So if it is golden, it's okay. Then we go there. But this is only a visual thing. I'm throwing into only one thing. But we use a software, say to a delta 2D, to find out exactly how much it is. You will define them one-fold change, two-fold change, three-fold changes. So normally for scientific publication, 1.5-fold and 2-fold changes is important. Is that clear for you, my colleague?
Okay, sir. Okay, sir. Sir, is there any thing which depend on the dye concentration? If the dye concentration is less or more?
That's another question. That's a very good question. For that, I need to go a little bit back. Okay. So okay, you might think that okay, this sample 1 might bind red at much higher concentration. Sample with this Psi5, it's less concentration. Concentration is same, but the binding may be different. This is the question. What we do is that we call as a swapping. That means, for example, every sample 1 will have 6 times. 3 times stained with Psi3, 3 times stained with Psi5. So it's a swapping. It's color swapping will happen. And then again the sample 2, 3 times with Psi3, 3 times with Psi4, Psi5. So that means we have treated each sample equal proportion with Psi5 and Psi3. So we have 6 gels. For sample 1, we have 6 gel images, each one with 3 Psi3 and Psi5, another 3. And then we'll have internal standard. Now it's called a swapping. It's called color quenching. The color quenching effect. So what will happen if there's an up and down when we normalize, when you take an average of all 6, then this dye to bind protein dye binding effect is eliminated out.
Okay, sir. Sir, my last question is that if we do the interlab comparison, so if my lab is having a result and your lab is also having a result, so if we match the result, then the result should be same or it should be different?
No, again with the interlab also we use same sample 6 times at least. Okay, so we take an average, and moreover we use the same protocol. In interlab also we use the same protocol, same staining method. So for example, if you are coming to me and if you're telling that, sir, I want to do this sample, I said, okay, send your sample, I will do perform the same experiment. So you are also doing the same set of experiment with the same dyes, same method, and same deduction methods, and same scanning. The scanning protocol is also there. So you need to pay attention for scanning. I will give 25 parameters to scan the gels. So you will use all this as a standard. When you have all this as a standard, then this differences is seldom. It's very, very little. Differences will be there, but it will be very, very little. You'll be negotiating. That's what we will do that, and we'll find out. I'll find out what happens exactly with the experience. If I see a gel, happen to see a gel, we can easily find out. It's easily going out.
So how much difference is acceptable? I want to know about it.
No, that it depends upon the sample. I will decide. It goes by sample to sample. It goes by sample to sample. But again, I'm taking for every sample 6 times from you and 6 times from me. That's 12 times. I'm going to take an average. Okay. So I'm going to take an average. So even if there's a small difference, it's nullified, automatically nullified. And then I will also do the background clearance. So in the software, in this software. Okay, so in this software, what I do is that I just take a spot. Okay, I will take a small region. I will ask this thing. Okay, this is the background color of this cold gel. Please clear it off. The software will do that thing. That's why the software is 10,000 euros. The software will do very clear. Okay, this much pixels I have removed from the gel. Okay, so all these things, all the informations, whatever I do, I put it in this 2D gel database. That means we are open. We tell that, okay, this is how it goes.
Okay, sir. Is there any open source software for demo purpose for the student?
No, I did not listen to the question. I'm sorry.
Sir, my question is that, is there any open source software so that we can give the demo for the students?
The open source softwares are there, but I don't use them.
Okay. So the result of the open source is not.
These are the softwares I recommend, because open source softwares, at the end I need to upload in the database. The open source software, if it has a bugs, I cannot find it. Here I can call the decoder on, hey, I'm calling so and so, tell me my answer. They have to answer it. But open source software is a kind of thing, unless you develop in your lab, you will not have the complete control. So I don't use it. But you are free to use it. As long as you know what is happening, you can use it.
Okay. Thank you very much.
Okay. So can I proceed?
Yes, sure, sir. Sure.
Okay, good. So now we go to the blotting, and I'm not going to explain about this slide. You run a gel, transfer to a membrane. It can be a protein, it can be RNA, and then visualize. That's all. So this what is what, and the visualization can be done with 3 ways. One is radioisotope. This is what normally the colleges used to teach. The radioisotope is no more used because of the hazards. The second one, we use a chemiluminescence, or we use an enzyme. The third one is an enzyme. The chemiluminescence, we use ECL kit or other things. And TMB is the system that TMB is a system we use it for. So can you relate this to the hair dyeing? Sir, from blotting you are talking about hair dyeing. Can you relate our technology to the hair dyeing with colors and everything? The people used to wear the gloves to do that. It's the same principle, the same method. So what I do is that there is a catalase enzyme that break hydrogen peroxide into water molecule and molecular oxygen. This molecular oxygen is used to convert the benzene, the colorless one, to the colored one, and the same technology used in the blotting, in the hair dye. So that's the principle of hair dye. Once I was working for L'Oréal Paris, and I was helping them in getting the answer. Again, the drug discovery aspect. So the same technology, we are using it for the detection. So with this, I just want to go into the different technologies which you know already. So we have southern, northern, western, and southwestern blotting. Other technologies are so funny. So we have southern technology, we have DNA, northern RNA, and western we got a protein. So with this background, we go to the western blot technology. This was developed by Harry Taubin, and that's why we use a Taubin buffer system. This is the western blot method. Burnett in 1981, they named as a western blot. It's otherwise called as protein blotting or immunoblotting. So with this background, you know about the protocol. You run the gel, transfer it to a nitrocellulose membrane, and then you get the signal. Then the signals is scanned, and you can also do the analysis. Okay, before I forget, one gentleman asked me, is there any open software we can do that? I recommend a software called as ImageJ. Please note down, ImageJ from National Institute of Health. NIH is freely available. You can do it for 1D analysis and 2D analysis. If you have any questions later, also you can email me. I'll give you the guidelines, and I can give you an online teaching. Okay, very good. So what we do is that we transfer it, and then we get the image. So again, primary antibodies, secondary antibodies. Sorry, I'm not going to tell you. But what is happening is that you have a sandwiched method and you have an ordinary direct method. In the direct method, the primary antibodies directly bind. The primary antibodies has a signaling in it. So you get the signals and you get connect. In the indirect method, we have a primary antibody, and then antibodies produced against them, and it has a signal. So those signals can be used for the detection of the things. Anyhow, I will share the slides. You can do that. And this is the protocol. I'm not going to going to deep into the protocol. What you need to tell is that it's a primary antibody. You can see the 6th point. We can dilute it in a range which is effective. Second point is that go for the 9th point. This is a secondary antibody which can dilute it 1 to 15,000 or whatever it is. And then finally, you detect it with whatever it is. So Western blotting steps is very simple. Again, like the proteomic steps, it's a separate, transfer, detect. That's all. So separation, you based on the STS page or native gel or 2D gel electrophoresis. You can do any technology. STS page, native or 2D gel electrophoresis. Then transfer it. The transfer can be, and again, it can be on the nitrocellulose membrane or PVDF or nylon. So use of these 3. And again, you can do the transfer passive. You don't have any equipment in the lab. You keep the gel image. You can keep the membrane. You can keep the filter paper. Keep the weight on that and leave it in the overnight on the table. Don't allow it to dry. That's all. Your protein gets transferred to the membrane. That's all. This is a direct method. It's a passive method. No electricity is involved. And then there are active methods, wet and semi-dry methods. Okay, this is the electroblotting explanation. This is what I want. If you keep assembled like this, so that means you have a filter paper pads, and you have a gel image which is separated It may be a STS page, it may be a 2D, it may be a Deutsch. Even Deutsch can be done. So if all these things is transferred, and then you get a membrane, the membrane can be a free nitrocellulose, PVDF, or nylon. And then you keep them, and then keep the filter paper above it so that the absorption will happen, and then you keep a weight on that so the transfer will take place. This is called as electro-blotting or electro-transfer methods. And as I told you, there are 3 methods: wet or tank transfer. It's like an electrophoresis. You have a solution, and in the solution, you put a plus and minus you apply, the proteins will move towards the membrane, and it fixed in the membrane. Otherwise, you have a semi-dry, which it requires, for example, this requires only a minute, say, for a wet or tank method, you use 500 ml of the buffer. Semi-dry, you will use only 50 ml of water. Then there's a dry method, which just take 5 ml of the buffer. So from from half a liter, it goes 50 ml. From 50 ml, it goes to the 5 ml. So that's what we call as wet tank method, or semi-dry, or dry methods. Again, these are the equipments. As I told you, you need to pour the solution, and you need to do that. And you have this cassette. That's what I explained. You have a cassette, and then you will transfer it. You can see that semi-dry. This is ECL semi-dry method. This equipment I have shown you from GE Healthcare. It's very simple. You have an assembly, the instruction here about the machine, and this is the first electrode, this is the second electrode. You can lay down 6 gels at any given time, and you can transfer 6 gels at any given time. This one is a complete dry method. It's an equipment which can give you very direct transfer and those things. So this is how we transfer it to the membrane. Once you transfer it to the membrane, and you can use 3 types of membrane. This is a choice of the and you will read that, so how it goes, but only thing is that the membrane size is 0.2 micrometer or 0.45 micrometer. So normally I prefer 0.45 micrometer because it's easy to handle and everything. Okay, so this is there are 3 membranes you can use it. It's your choice. And the transfer buffer, the first buffer is the Tobin buffer, then CAPS transfer buffer, and bit stress buffer. So depending upon all this system, you will get it. And we use a methanol. The methanol is used in the transfer buffers because this will make the protein to easily transfer. That's why the reason for this one. You can use the buffer system. Most of the laboratory we use a Tobin buffer system. And then what we do is that if you want to check, this is the protocol, all the steps. You have a cell lysis, your quantification of the protein, separation on SDS page or 2D or native electrophoresis, and then you transfer to the PVD membrane, and we use a Pondicherry. This is a temporary qualitative staining. It's not going to stain it in quantitative way, just to see that if the transfer has happened. Then once it's done, then you treat with primary antibody and secondary antibody, and you visualize it. So again, the treatment I already highlighted. There can be direct method, where direct method has an enzyme or a radioisotope or a chemiluminescence signal. So these are types of signal: an enzyme or enzyme may be horseradish peroxidase or alkaline phosphatase, and then it can be a chemiluminescence which can give a signal, say luciferin system, the oxyluciferin and luciferin system, or you can go for radioisotope. Radioisotope is an old technology which we don't use anymore. Even in your teaching, please avoid that. You tell the students that this radioisotope is no more used because of the health hazard. You need to have a license for example, in India, we need to take a license from BARC, Bob Atomic Research Center, and then you have to use it. So without an atomic commission certificate, you cannot use it. Then, these are the 3. If you use only one antibody for detection, it's called direct method. When you use a primary antibody and an antibody to that, we call a secondary antibody. And the secondary antibody has got signaling molecule, the same signaling molecule. That is called as indirect method. So the direct method advantage and disadvantages, which I'm not going to do because it's very easy. The Western blot, what we do is that after transfer into the membrane, we need to block the things. The blocking can be done with the skim milk or milk powder, which is normally from the shops. Or normal serum we can also use, or use highly purified proteins. Highly purified proteins is accurate, but the answer is it's very expensive. So we have a blocking buffer. You can test the blocking buffer, and ideally the blocking buffer should fix all the region, because this membrane, nitrocellulose, PVDF or nylon membrane, they can take up protein in any region. So we want to block it. We want to avoid everything, and then we can do that thing. There is commercial blocking blockers also. Again, it's expensive. You can take it, and then you have a vast buffer system. There are several buffer systems, say TBS, PBS, TBST, or PBST. You know that what is what, and then pH is different. You can use it. You can choose the blocking buffer system, and you can go with the experiments. And then you have a primary antibody and secondary antibody. I told you that, this is for detection technology. You can really test it. You have to optimize it. Normally you will optimize it. What's the concentration of primary and secondary antibodies depending on the concentrations, and then you will get it. So detection method, I told you already. We have radioisotopes are earlier used and now replaced. So that means it's very, very clear information. The enzymes are, you can use a chromogenic, fluorogenic, and chemiluminescence things. Within enzymes, we have oxidase, peroxidase, and alkaline phosphatase. And with the oxidase, peroxidase, I don't need to give you the detection methods. There are several detection methods. You can see in the laboratory. You can stop it. You can and you can also use this for the chemiluminescence also, and you can scan it in the GLDOC system. So normally, what I do in my laboratory is that I use chemiluminescence. So I use the chemiluminescence ECL kits. What I do is that I put them on the scanner, say the GLDOC system. I'll take it in 1 minute. So 1 minute, 20 pictures. That's what my criteria. Take 20 pictures in 1 minute. Take the membrane out of the machine. Put them immediately in the water to replace all the excess reagent, and then go for TMB buffer. That means the second detection system, which is physically visible. You will see that. You will see the spots. You will see the bands. That's very clear. The detection method, I do it two times. One is ECL, other one is my own the TMB buffer system. Then now I have an image in the computer, as well as I will have an image of the same blot itself, and I can scan again using my own paper scanner, and I will get the best image. So most of the people will wonder why Western blot is just such a difficult technology. How you people are doing? So there's a high success. The success rate is high because we optimize every steps. So this is how we do that. It's the real things. So the Western blot visualization, again I told you, it can be chemiluminescence or HRP, the oxidase peroxidase labeled secondary antibody. Okay, so these are the blots. I mean, this is again from one of my PhD students did this things. What we did is that we have two types of Brucella abortus and Brucella melton. Brucella abortus is a microorganism that affects the cow and buffaloes, and Brucella meltons affects the sheep and goat. But if you look at the genome, they are same. The genome is same. Why the host specificity? So that was the question we have. So we got the serum from cow, buffalo, sheep, and goat. And we ran it as a primary antibody, and we cut some of these bands, and then we were able to successfully identify those bands as a protein. And we found that they are very, very good. And we just go into that. And that's a list of the publication. You can go through that publication and history. Anyone can download it. It was in 2015. So again, in the Western blot, when you have an up and down regulated proteins, you also need to talk about housekeeping proteins. How will you look at the housekeeping proteins? The housekeeping proteins are the one which is supposed to be not influenced by your experimental conditions, but in reality it doesn't happen. It will be influencing. So what happens is that you need to decide what kind of housekeeping protein you need to have. And again, troubleshooting, I'm not going to deal everything, but troubleshooting means there are a lot of challenges you will get. I will supply the slide, you can read it and you can get it. For example, weak bond or high background, so what you want to do it. And these are the hall of shame. That means you can run several. This is from the University of Rice, where they have a hall of shame. In my laboratory also, I had a hall of shame, but I don't have a better picture. You can see that when you're running a gel, you will get all these funny things. So you will end up with all the things. That's why this troubleshooting is very important. As a professors, as a teachers, you need to know more about troubleshooting than the technology itself. Once you know the troubleshooting, then you can really answer this. Okay, the Western blot seems to be complicated. You see that you have to do the I'll go back. So you have to do all the steps, detection and everything, labeling and everything. So that's all complicated. So what we do is that we have a new methods which is called as automated Western blot. It's called as hands-free automated Western blot system. We also need to modify ourselves. We also need to go for that. So you can see the physical method. You have a blocking membrane, you transfer, block the membrane, incubate primary antibody, multiple washing, incubate secondary antibody, and multiple washing, and then detect. So this takes a lot of time. So in order to avoid this, we have a lot of companies, and I take the Thermo Fisher company. I'm not marketing Thermo Fisher, but I'm highlighting that automation is possible. So there are a lot of other companies also. You can free go into the things. Again, I'm not marketing one company. I'm just highlighting that the Western blot can be automated. So that means all the steps, washing and everything is a very tedious step. All the steps is bypassed with this machine. What we call as the bandmate or iBind technology. They go for detection. So you can see that again, the iBind system, the detection is very quick, it's very fast. But if you do it traditional Western blot, it will take a lot of time for the detection, and then it will go things. And I really recommend you. There's a paper Misra, Tiwari, and Gomes. That is in 2017. It's a review article in Expert Review. Protein purification and analysis, next generation Western blot technology. So you can know more about this Western blot for your practical purpose or for your real things. Okay, next comes very thing. Now we know what is 1D SDS page, what is 2D, two-dimensional electrophoresis, what is Western blot. So in all these things, you find a spot, you find a band, and you are interested. For example, the night gel, I told you an example, this green spot and the red spot. This green and red spot, I want to identify what is the protein behind that. So how I'm going to identify? So I'm going to cut this protein. Once it's cut, I'm going to subject them to the trypsin digestion. Then I'm going to put them in the MALDI, mass spectrometry, and then we get a spectra, and this kept compared with the database of NCBA and Swiss plot. It's an automatic process. I'm going to show you how this automatically we are going to do the other things. But let me just see the time. Yeah, I think we can able to do that. So what we do is that we just go for PMF. It's called peptide mass fingerprinting, or MS or tandem mass fingerprinting. Don't worry, I will explain all these things in coming slides, and you will get an information like this. From the decider or from the software, we get there is upregulated, downregulated. From the software itself, you are labeling the number of spots. So these two columns you are getting from the software. The list of the columns, name of the protein, MS score, we call as mouse score, molecular weight, PI, peptide mass, and coverage. All these things you are getting from the from this experiment. So this is how we do the protein identification. So I repeat. We cut a spot or a band from SDS, spot from 2D gel electrophoresis, or the Western blot membrane itself. Then you go for we are going for trypsin digestion of the proteins. And then you are going to spot it on the MALDI target plate, and you are going to get the MALDI spectrum. This can be compared, and you get these things. So before going to the MALDI, everyone knows that MALDI, but anyhow, I want to tell you how this origin. So you have a steel target plate, or you can also have a gold target plate. So it can be anything. So you have a matrix. Matrix is an UV absorbing substances. You will have 1,000 molecules of matrix and 1 molecule of protein. That's why you can see that there's a random protein which is here. So the matrix concentration is very high. UV, yes. Excuse me, someone has opened the mic, and you are supposed to open the mic only to interact with us. Can you close?
Yes, sir, it is muted.
Yeah, please mute the mic, because when you are interacting with outsiders, you don't disturb the others. Okay, good, very good. So what we do is that we have a steel target plate, and we have the matrix. The matrix has UV absorbing substances, and we have a protein scattered here, and it's a mixed here. I will tell you how to do that. And then we have a laser beam. It's a 337. It goes as a pulse. It goes as a pulse. For example, 40 laser shots per second. So that's the speed I give. And then what happens is that the UV absorbing substances matrix will pick up the energy because it's already UV, and then it melts. And during the melting, it also melts the protein which is next to that. That's why it's called a soft ionization. We are not directly exposing the protein to the laser. Otherwise, the protein will get chapped. It will become like your dosa. It will become brown. So we don't want to do that. We just want the matrix to melt, and the matrix melting, it melts the protein, and the protein gets ionized. That's why it's called as matrix assisted laser desorption or ionization. You might ask the question, why desorption or ionization? The concept is that we really don't know what it exactly happened. It's a desorbing or ionization. So it desorbs and then ionization. So that's what we call MALDI. So once we get the MALDI, so once I took this whole stuff into the one line. This is the target plate with a sample on that. So you have the laser hits. Then you have, for illustration purpose, I have got only 3 types of ion. But in reality, you will have 100,000 ions for every protein. 100,000. That means 1 lakh ions will be coming. What happens is that for illustrative purpose, I just take only 3 colors. One is green, other one is blue, another one is red. The red one is bigger one, the green one is medium, and the blue one is smaller one. And once the ions are produced, generated, then we have an accelerating pulse that pushes this whole ions into a vacuum tube. The vacuum tube, it may be a linear, it may be 1 to 3 meters. So it can be 1 to 3 meters. It's a vacuum tube, and these ions are drifting. We call it a drift region. It's a drifting, and again, the speed of this ion is dependent on the mass by charge ratio. That's what we call as M2Z ratio. And we have a detector. Detector detects, detects these ions. They also detect counts. How many ions from each category is hitting? I'm telling about 100,000 ions at any given time. So all 100,000 ions are detected the deductor. And it's the deductor will convert this into is a digital information, and digital information is something that you will get an graph. So for example, the blue ones are 4, and they're higher in number. The green one is only 1 and red is only 3. So what happens is it goes here, you get intensively top, because we this is MSET value of 4,000. At the same time, green one is a bigger one, it's 6,000. You can see the relatively smaller peak. The red one is called as 8,000. So this is relatively bigger. So this is 4, this is 3, and this is only 1 molecule. That means intensity depends upon the number of ions, each ions. And intensity, if you look at this x-axis, we always have an MSET value. It can be a nanosecond also. We can also convert them into nanosecond. But intensity, there's no unit here. It's an arbitrary unit. That's why I specifically leave this empty. Arbitrary unit. It gives you the intensity. So this is how we get the spectra. And if you look at this, you have a MALDI here. I go to the previous slide. So I go to the previous slide so that we have a MALDI on the thing. Once you get a MALDI, you are separating in a vacuum tube, and you are calculating the time of travel of each ion from this point to this point. That's what a time of flight. So you get MALDI of MS. So that's what the reaction MALDI of MS. We have complicated systems. I will show you in coming slide. So this is the simplest way you get a spectra. And once you get a spectra, you can do a lot of things. Again, the matrices, there are a lot of things depending upon the nature of the substance. You are using proteins or sugars or oligonucleotides or genes. You can do whatever you want. There are a lot of things. My favorite is alpha hydroxycyanosinamic acid and DHP. These two are the things which I normally use in the laboratory. Of course, I have all the reagents, but I use only these two because that gives a very big advantage. Desirable characteristic, I'm not going to read it because everyone knows that. And again, the multi-lasers, there are different types of laser boxes, and each laser box has got its own things. And normally, we use the 327 nanometer nitrogen beam. So most of the multi machines, we use only this one. The rest of the things is for special purpose. Okay, with this we go into the spotting. The spotting is very simple. Once you triptych digest the sample, what we do is that we go for dry droplet. That means you have a multi-target plate, and we just drop it on the target plate. You mix the matrix in equal proportion. So here the matrix, say 0.5 microliters of the matrix solution and 0.5 microliters of the sample. You mix them and drop it and add dry. That's all. Very simple. And there's another one, surface preparation or add-on. This is a very good technique. You have salts and everything in the sample. So we spot the sample, say 0.5 microliters on the matrix or multi-target plate, and then you wash them, say with bicarbonate. You can wash it with bicarbonate, sodium bicarbonate, 2 or 3 times, and then you add the matrix on this. So that means in the previous case, you mix the matrix and the sample, drop it on the multi-target plate. So that's called as dry droplet method. In the second case, you add the sample, wash the sample, and then add matrix. It's layered. So that's what called add-on or surface preparation. So you prepare in such a way. These are the two basic methods for the multi preparation. And the advantages is relatively you can go for 3000 Dalton and you can really do a lot of wonders. And disadvantage, the matrix will give you signal and photo degradation because you're using a laser, and the matrix solution itself may create a problem. So these are the 3 disadvantages. Matrix solution react and give a background signal. First case. Second one, the laser can give you photo degradation, which you cannot trap it. The third one, the acidic matrix will create changes in the protein solution. These are the 3 ones. So with this, how we are going to do the protein identification. From this slide onwards, please pay much attention because I'm going to explain you with example how it is going down. So we have taken a myoglobin structure, the myoglobin sequence, and we are going for triptych digestion. You know that trypsin will go and lyse it or break the proteins at lysine and arginine. There are 2 positions we can do. So mostly lysine and arginine, it can go and cleave it. So when you cleave it, it will give. It's like restriction endonuclease. There is a protease inside. So that's the difference. So what we'll get is that you will get a different mass points and you will get a different peptide. So the whole myoglobin is split into, you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 peptides for the example, and you will get a spectrum like this. I told you before getting into the things, I want to explain here one very, very important thing. Speed is something like this. You're asking a rabbit and an elephant to run across the forest. The rabbits are smaller, they will go faster. Same thing here. The smaller the ion or the charge, they will go faster. At the same time, the elephants, they're big ones, they will go slow. That's what exactly going here. Okay. Yeah, that's what exactly going here. The smaller, you can see that 748.43, this is coming here, and then the rest of the peptides, it goes like this. Second one is 1502.66, so it's somewhere here. So and then it goes like this. So all these things will be separated on the things. Again, how this identification is done? I told you that it's matching with the database. The matching with the database is very clear. So you have a query spectra. So these are the masses which you get in your spectra. This is the database. Database can be NCBA or UniProt or Swiss plot. We call it Swiss plot earlier days. Now we have UniProt, the same thing. So what we do is that this will go and match this peptide mass. That's why it's called as peptide mass fingerprinting. So it takes the peptide mass and you do the fingerprinting in the database. EMF. So you get the peptide mass fingerprinting. So you get two masses, about two masses of known one. One hits this one and 3 hits to this one, and there are 2 masses unknown. So conclusion is that 3 hits is here. Naturally, this could be the protein. So this is how the assumption is done, and this is how we get it. The problem with this is that BMF principle is a wonderful thing. You need to understand something very, very clearly. So I told you it's already digested with trypsin. Trypsin is not the only enzyme. You can use several other enzymes. Chymotrypsin you can use a chemical cleavage. You can use anything. So, but you need to know where this cleaves. For instance, to understand the BMF principle very clearly, I have given 3 proteins: protein 1, protein 2, and protein 3, and each has got the same sequence. Same amino acid, same number of amino acid, and the content is same. And if you look at the molecular weight, they are same: 4842.05 or 4842.05, whatever you can pronounce it. So again, you can see the 3-1. But again, if you are treating with trypsin, it gives you 4 peptides, but different sequence. You can see that this one, K is here, K is here, K is here. So it will give you a peptide like this. And for this one, it will give you a peptide like this. And this will, when you go with the same thing, so this sequence, or you see this 4 peptides is given, that means you need to give 4 signals on the peptide. But in reality, you will get thousands of the molecule. But in here, I just for explanation purpose, we have taken only 4, and you can see that how this 4 gives. You can see that the whole spectrum and visible eyes itself are different. Same thing, you can see that the same molecular weight, they have different sequence, and trypsin digestion, different peptides, and then you go into the things, you get a different things, so that you can really see that they are completely different. The identification is highly specific. This is the basis of your peptide mass fingerprinting, PMF. So now you know what to do and what is the sequence. So similar things can be done, and most of the popular software is that Matrix Sciences. It is free software, anyone can use it. So this is the software where you can go for peptide mass fingerprinting or sequence tags or MS. I will explain what is MS. Before going into the peptide mass fingerprinting or explaining the peptide, I'm not going to read the slide, but only information is that it is like thumb impression. Each protein has got its own sequence. That's the only carry home point. That means you can separate and identify each protein specifically. So your question comes. So here your question comes, sir, if the protein is not in the database, you mentioned in the first slide 84% of the annotated human protein is there. What about the rest of the proteins? Because we know only 84% percentage of the proteins in the human database. But what if I get in the rest of the 16%? That means they don't have information of this in the Swiss plot or UniProt or NCBA or DDBJ. You can also use the Japanese database. So all these things don't have information of your single protein. So in this case, you go for a special technology called the de novo sequencing. This is applicable for mostly unsequenced organisms. So there is an unsequenced organism, you can get it. There are a lot of software to do that. For example, BioTools from BDAL, this is Google Daltonics, Nova, Peaks, Ludwig, Nova HM. So many things are available. So these are the handful of the softwares which you can do that. Again, the PMF on the web. So I gave the database search and de novo sequencing. There's understanding the spectrum from an unknown sequence. You can do that. So this is what use it for the second case we use for this de novo sequencing softwares. And for the PMF, you can use a lot of softwares. You can use Mascot, which is my favorite. Found, mouse, peptide search, peptident. It's not pepsodent. It's a toothpaste. Here is a peptident. So you will use that. So PMF algorithm, I'm going to do. I have already explained. I'm not going to do that. And the Mascot server, it gives like this: the peptide mass fingerprinting and sequence query. This is the server. This is how your website looks like. The Matrix Science. You can log in. We can see that if it is possible to share now. Then we can also have the same thing, and you can click search. You can here you can go Mascot. You can see that Mascot data search. Click it, and then it gives you 3 access. You click this one. you upload a database and you will get a result like this. Whichever is coming with the p-value, you can see that log p-value and all these parameters. You will get a protein, and this protein is identified. This is how we identify the things, and you will get the results like this. If the organism is not sequenced, then you need to definitely go for de novo sequencing. That means understanding the peptide. So the limitation is that you know that some limitations are that I'm not going to do. But again, you go back to the imagine the same myoglobin you are taking. You're getting the peptide. Can we further break down this one? That's the question we have. So the answer is yes. Can break down, and that one based on that we have two types of machine, what we call as linear multi and reflector multi. So I will go into this one. So before go explaining what is linear and reflector, I'll explain this one. So you have a mass spectrometer, you have a PMF spectra. These are original spectra which was generated by myself. So you have a PMF spectra. Once you have a PMF spectra, then you lift one of them. You are going for MSMS. That's what called as tandem mass spectrometry. So you lift one of them and go for these things. You split. Now this was peptide. Now you are going to the protein level or amino acid level. So you get the amino acid level. Again, you compare with the database. This one, this MS spectra, is also not available. Then you go for de novo sequencing and you get the protein sequence. So this is how we do the things. So you run a scanning, full scan. Then after full scan, you compare with the database. If the information is not available, it's unsequenced organism. Now you go for MSMS, and then you get more things. And MSMS is also done to increase the confidence of your research. So you get the more information. Then you go for de novo sequencing, and then you go for the peptide identification. So this is what I want to tell you. You have the laser box, you can camera and light, so you can see everything. All the spots can be seen. For example, it's a 96 well plate, like target plate, or 364 target plate, question target plate. You can see every spot. And then you have a laser box. The laser box will kill the laser, and the ions are coming out. We have an electrical energy to push this into a vacuum tube. Vacuum tube, it passes to the first detector. It's called as linear detector, or it is reflected to the second detector. It's called as reflector multi. Very simply explained, you have a ball in your hand. You're throwing to the wall straightly in your straight facing you. That's called as linear multi. So you have one detector. So you have a sample. Imagine your ball, and you have a vacuum tube of 1 or 3 meters, 1 to 3 meters, and then you have a detector. So you throw the ball, the detector gets the signal. On that one, it's called as linear multi. So you have one multi TOF MS. That's the information. Multi TOF MS. And then in the wall you have a reflector. Say you have 3 balls now. You have a green ball, red ball, and a yellow ball. And you're throwing them. You are asking the machine, I want to choose only the red balls. So what will happen is that I throw all 3 balls. The 3 balls will travel at different timing because of their MSET value. That's mass to George ratio value. So they reach the detector. In the detector, I have already told the machine, please reflect only the red ball to the ball. Detector places in the backside of myself. So the red ball goes there and gets reflected. That's called as reflector multi. So now we have in the second reflector multi TOF MS TOF MS. That's why we call it as multi TOF MS. This is what you see in the literature, or this is what you see in your book. So this is otherwise called as tandem mass spectrometry. With this background, if you look at this explanation, you will understand what is linear multi, LT and RT. So linear transmission and reflector transmission multi. So these are the 2 types of multi machine. In the linear transmission, you will get a peptide. In the reflector transmission, you will get an amino acid level information. Once you get an amino acid level information, you can really do that. So the explanation is like this. So you can see that I have 3. Listen carefully to the tandem mass spectrometry. It's called as multi TOF MS. So you have 3 peptides: GD, PHR, HED, and something. You are passing this in the mass analyzer. It reaches the detector, and you are getting 4 peptides versus 1 non-specific peptide. So now 4 signals you are getting. What we are trying to do is that now we are using the second scanner. That means you are going to the wall. In the wall, it gets reflected, and we choose only HED. This HED is getting fragmented. It's fragmented in different angles, and it goes in the mass analyzer. And you get to detector, and you get a PMF spectra, sorry, MSMS spectra. The previous case, this is PMF spectra, right? Mass fingerprinting. You have only 1 mass analyzer, but this case you have 2 mass analyzers, and then you get what you call as the MS also, and then you can go for de novo sequencing and you can get what the sequence exactly. So you know now by two methods, BMF and MS. And if you want to know about MS, you need to know about the molecular masses of every amino acid. So each amino acid has a 3-letter code and 1-letter code. You need to know what is that, and you need to know about the masses. You can see that masses is 5 digits. That means the moiety can specifically go up to 5 digits. Give me a second. It can go up to the 5 digits. That means it's such specific information. You can see that de novo sequencing means you are understanding something. De novo is applicable in every field. Law, it is applicable in biochemistry. De novo means purine pyrimidine biosynthesis. So law is applicable, commerce it is applicable, pharmacology is applicable, so many things. But here de novo means, de novo sequencing means understanding the spectra and getting the sequence information. For that you need to know what kind of ionization takes place. The fragmentation can take place because of the ionization, because of the laser. The second one because of the acceleration. Third one during the flight in the drift region. So these are the 3 different locations you can get it. So what we do that again, I told you how to do that things. This is the slide which I want to highlight it. You can see that there is a peptide composed of 4 different amino acids, and these 4 different amino acids can be split into different things. A, B, C ions. So you can see that if it can split here, this is A ion and the rest is B ion. And again, if you look at that, the A, B, and then other side of the things, it can also split here. So you have X, Y, Z ions. So there are 3 types of for 4 amino acids, you will get minimum of 12 ions. So sorry, not 12, it's 18 ions. So 9 XYZ, 9 ABC. So you need to understand with this, and you need to know about this information also. Then only you can show what is B and what is Y ion. So this is what the peptide fragmentation goes, and then you need to know what is A ion, what is B ion, what is X ion, Y ion, Z ion and C ions, and then internal ions. If you know all these things, then yeah, again this is the ions which to explain this one. You can see that you can go for this one. Pay attention now. You have a DNA fragment, protein fragment. This is the fragment, and this fragment can go for the peptide clearance like this. So you can see that this molecular weight is 88, and this Y ion is 147. 147 or 175. This is purely because of your arginine and lysine. So this is the trypsin cleavage. So the first amino acid should be K. You can see that the first amino acid, the last amino acid is K, because that is where the trypsin gets cleaved. So you can see that this can split something like this. You can see 88 is B1 and Y1 is 147. I did not mention A ion, B ion, C ion, only B and Y ions, because these are the predominant ions you need to see in the spectrum. Once you see the spectrum, then your answer is okay. Now the same peptide. So I took this peptide, then I theoretically show you what happens exactly. You can see that the Y ions are here. So pay attention. The Y ions is clearly visible to you now, and I'm going to show you the B ions. So the Y ions and B ions are distributed here. You need to look into the spectra carefully. One of my colleague, Dr. Chris Weiss from Free University, he's a very, very senior professor. He used to sit in the computer like this from whole day. You can't imagine how, and then he will write the whole sequence by heart. I used to use a software. I mark it physically. I mark physically and get the information. So that means if you know this A, B ions and B, Y ions, then you can really get these things. So this is one of the spectra. You need to get a good spectra for doing this one. You can see that lift span, see the heading, lift spectra of 2320.350. This was published in 2007, I believe, some 13-14 years before. That time we did the de novo sequencing, because this was from a crab, and I was able to successfully mark it. This was all physically marked. This was all physically marked in a software. I marked one by one and I got it. So for a sequence, I worked on 1 week to get the sequence. That was a beautiful story. And the same swing, you can see that 2320, same thing. It's not a good spectra, because there may be detector error or something like that. You need to have multiple measurements, and you need to get a good spectra. You can do that. At the end, the protein identification can be done in 2 ways. One is that you can go for PMF and database search. If the database doesn't have anything, you need to go for the de novo sequencing. So these are the things which I want to highlight. And with this, I also want to use this, since you are all of you are biologists. Our university is open for PhD students. Anyone, arts, BA, science, engineering, management, they can apply. At the moment, we are offering 20,000 rupees as a stipend. And we also give on-campus free hostel and mess. That will cover another 15,000. Food and hostel is provided free. We also provide financial support for the international and national conferences. We also support publication charges and patent charges. I think patent spelling is wrong. And our whole PhD fees is only 10,000, only one time. There's no other fee, no examination fee also. You will appear for the examination, you will appear for defense, everything. Thesis submission, everything is go. There's no entrance examination for those who have got DRF and NET. If any of your students are interested, they can refer our website. I'm also the research coordinator of the university, the whole university. So you can write to me or simply research at SRM-AP. I will receive the email and help them. So this is the open PhD position, and we also have a faculty position. At the moment, we are looking for bioinformatics, neurobiology, and other things. The only condition is that they should have a master's and PhD in bioinformatics for the bioinformatics position. And they should be able to handle multiple projects, collaborate with internal and external universities. And for all other positions, we need either an abroad degree or a primary institutes of India and an outstanding research award. You can directly write to me, or you can log into this. So with this, we finish this session. If you are having any question, I can answer. Before that, what I want to do is that I want to stop sharing this one. How to stop sharing? Okay, I have to stop sharing. And then I want to share. Give me a second. I'll come to the question section very shortly. Give me a second. I will explain something, and then I'll come to question section. Okay. Mascot search. I want to show you physically. Okay. That's what I want to say. How to share that? My window, and then I could not see the browser of mine. It's there. Give me a second. I'm sorry for the delay, but I'll get it. Okay. So window. Yes, I'm able to see that. Okay. So we will see the matrix sciences, how it looks like. I think you are able to see my. So I just picked matrix sciences, the PMF, and I go for the database. Okay. I click this. So this will open something like this. I told you in the things. And I click this one. So it says access mascot server, if you have a server, or peptide mass fingerprinting, PMF, or sequence query, or MSMS. MSMS is the reflector mode. We will go for this one, perform search. I'm doing live now. So I just click it, perform search. It will open a website, and then you need to do the things. So this one is okay. I give test as a thing, and I need to give my name. You also give to name, and then the email ID. You can do it yourself also. Then I want to take which database. Just want to take, say, any database I can take. I just don't want to go in. I go for the Swiss plot, this otherwise uniplot. Then I use a trypsin as an enzyme. As I told you, there are several choices of the enzyme. You can choose whatever the enzyme you have got it. But I use a trypsin, and I allow one missed cleavage. That means there may be mistakes in one. And then now I want to go into the database. Which organism I want to choose? For example, human or animal or whatever the organism you get, or plant also. For time being, I just take everything. I told you there's a urea treatment. The carboxyl methylation is a fixed modification. So by experience, I just take it. And variable modification, all the methionines will be oxidizing. We'll check. These are the different modifications available. I have to go to wait. Give me a second. It will keep like this. Yeah, oxidation methionine. So oxidation methionine, just click it. So I gave, and I clear all these things. So every machine has a tolerance. So I will just clear tolerance. I just take the default now. Then I browse the things. I have some files. For example, I have a protein digest. Say randomly I take one file, and then I open it. So that means my file is added. So this is from the MALDI data. Once I get MALDI data, then I go for the things. And then what I do is that I start search. That's all. The system automatically goes into the Swiss plot. So it's online. You can see that it starts searching. So searching is run. Mascot terminated with error. Okay. Now I understand what's the error, because we need MSMS. We have taken the MSMS spectrum. That's why it says error. Same procedure. So I give and then I use only Swiss plot. I don't want the contaminants. I just throw the contaminants out. I use the Swiss plot, and I give all entries. And then I don't want to go for any other thing. Enzyme is trypsin. Okay, and then the fixed modification box immediately. So I take it, and for this one, I want to go for oxidation. So oxidation is methionine. I just take it, and then this file Google give it the data file, because it's an MGF file. It has got a lot of spectra. So I just search it, and then I click search. That means now I have given a MSMS search, not PMF, but it works on the same way. You can see that it starts searching, and you will see that now it can see that it's Wednesday, July 14th, 8:55, because of the US timing, because the database is sitting in Europe timing. The database is sitting in Europe. I can also choose Japan. That will go in advance. And what we do is that it searches. So now it's 84% covered, and we'll get it very soon. That's a wonderful thing. Okay, so it's calculating. It's done. It gives you the results. Yeah, now I got the results. You can see that. So this protein is something like this. And what I want to do is that I can also go for report builder, or I want to what kind of protein is this. So I just click this one. It says that this is a protein adenine transferase. And if I click this, it will open another window, show me the spectra, and it will show you the X ions and Y ions, how this protocol has been calculated. You can move around, you can zoom, you can click it, and you can find it. And you can also go for all the ions. I told you A, B, C, X, Y, Z ions. And once you know that validation, you can do that. You can also save this. You can also submit this to the data bank, because at the end you need to submit it in a data bank, and then that way your protein identification is fixed. This is how we do the protein identification and the things. Okay, now I stop sharing. I will go to the slide, and I'm ready to answer your questions. I hope it goes in a supersonic speed. I know that, but I give three different to summarize the protein extraction from anything, and then you go for the SDS page. You go for the Western blot. SDS page, 2-dimensional electrophorosis, and if you have an antibody, you go for the Western blot. And from there, you go for the cutting the spot. Analyze it, cutting the spot, and once the cutting, trypsin digestion, go for MALDI-based identification, either PMF if your information is provided in the database, or you go for de novo sequencing if it is not available in the database. Okay, thank you very much, and I'm waiting for your questions and comments.
Thank you, sir. Thank you for the energetic and informative session, sir. Now we are aware of protein sample preparation, protein separation, protein identification, and bioinformatics approach in the proteomics research. Yeah. We obtain knowledge in the applications of multitap, tandem mass spectroscopy also, and different softwares in the proteomics field. We are also pleased that you have shown a slide demonstration of mascot search, sir. Yeah. Dear participants, now the session is open for questions. You can ask the questions.
Don't hesitate. You can ask any question. I will answer it. Maybe in general proteomics also. You don't need to really ask whatever we present in the slides. And I'm waiting for your comments. You can also write your comments. I can see what's your comment.
Excuse me, sir. Am I audible?
Yes, you're audible, Dr. Surya Pratap Singh.
Sir, my question is that how we can know the protein-protein interaction by the use of this technique? Is it this technique helpful to know the protein-protein interaction?
Yeah, I tell you, that's the next level of analysis. We get, say, 100 proteins upregulated and 100 proteins downregulated. Okay, so we have other softwares to do that. Maybe I can little bit share again this window, show you what is what. So you see this window, right? So for example, you have a software called string molecular. I know this already, but anyhow, I'll show you. String. For example, this software. Okay, it's a wonderful thing. It has got all the information of this one. And what I do is that I just click search, and then I have an example here. I just take some examples from there, or from here. So you can search either by protein name, protein by sequence, multiple proteins. Multiple proteins is the one where you want to understand the sequence. So I take an example from there. So it gives 4-5 examples. And then it's from E. coli, but what I do is the software is that I just say autodidact. It detect, auto-detect. Then maybe some other thing. I don't know why it's not working. Maybe I have an issue with this computer, but anyhow, I just get these things and I click this. It says that these are the proteins which you have fed. It's only 4. Reality will feed thousands of protein. Okay. Then what I do is that I continue. So if I continue, then it will give you the protein-protein interaction like this. It's a beautiful. You can move that. See, you can move that. You can save them. You can go and download as you like. So you can view and download or export. Export is like whatever the image you want. Again, these colored images are everything information, and it is within this round. It's like a moon. You can see that there's a sequence, the 2D structure of the protein, and you can also turn it. You can also do whatever you like, and you can also go to the source. If you believe that this is too much, what you can do is this information is got from several things. Text mining. So here you can see that I don't want text mining. I want experiment. I want database. I don't want expression. I don't want neighborhood. I don't want gene fusion. I don't want expression. And I update it. You can see that the whole is small now. So you can look for what kind of information you're looking. The protein-protein interaction, you get it here. If you want to know this information, click it. It will take you to the PubMed itself. So it will link to this. This is not the only software we have. We have another software, say David. This is one of the software which I really like it. So it's a David software which gives you a wonderful thing. For example, here also is chart analysis. We can take an example from them itself. So demo list 1. I take a demo list 1. It's human proteins 149. And here it gives what kind of annotation you want. You just want the functional annotation. Click it. It will give you all the results with one click. And then you want to know about, say, the pathways. I'm interested in pathways. I'm interested in cake pathway. There are 66 protein out of 145 proteins. 66 proteins are interacting each other. This will open a new window. I'm not so sure whether this new window is visible for you. It will open a new window and it will show you the exact pathway. Is the new window is available for you? Are you able to see that new window? Hello.
No, sir.
Yeah. Then I will do it. I will stop sharing. Then I will go for sharing the entire screen. That should be clearly visible now. Share. Now, are you able to see the new window?
Yes, sir.
Okay. So, it's pathways in cancer. I'm interested in transcription misregulation in cancer. There are 6 proteins. I click this. It will open you. It will show you these are the proteins which is available there. And you can download. You can save it. And again, you can go into some other thing. For example, gene ontology. This is what of interest. So we just take, say, gene ontology, anything. So what I do is that I take this one, gene ontology. It will give you the same window. Then it will tell different related things. If I click that, it will open the whole thing, and gene ontology, it will take me to the GO annotations. This is what the information we get. Again, within this software, we can also go for, say, pathways. I'm interested in pathways. I just click again this one. I go for this one. Then I go for this transcription regulations. If I click this, it will give the things. But if I click here, it will open me the whole pathway. You can see that. Are you able to see that the entire pathway and the blinking ones are the protein from our interest? Are you able to see that there is red blinking? Yes or no?
Yes, sir. Yes, sir.
Yes. This you can save it and you can publish it. This is how we do. So the protein interaction is another concept which we normally work in a different area. I mean, the next level of research. So now we are confined to 2D, SGS 2D, Western blot, and protein identification. And how we are using this in the research. That's what I highlighted in the slide. I think I have to go back to that. Wait. I have to go back to one slide because that's the connecting slide. All the things. It's a long slide. So give me a second. I'll get it back. 1 second. I'm scanning my own slides. Yeah, I got it here. So, there. Yes. This is the slide. So you can have a drug, you can do the sample, extract the protein, you can go for biochemical analysis, you can go for proteomics where you use gel, SGS page, Western blot, and then you go for this one. And microarray, it did not cover it. Okay, so we'll continue the questions.
Dear participants, you can ask the questions.
Sir, can you show that slide once again?
Dr. Surya Pratap Singh, I could not listen to you. Maybe you can repeat this.
Sir, he's asking to show the slide once more.
Oh, you want to see the slide once more? Is that right?
Yes, sir. Yes, sir.
Okay, that's not a problem. Okay. Is that okay?
Yes, sir. Yes, sir.
Good. So, any questions?
Sir, my last question is that if we are missing some amino acid in our protein molecule, then is it possible that we can rebuild the protein and how we can know the missing amino acids in our protein?
You mean you want to mix the protein and amino acids?
No, sir. My question is that while we start the docking or molecule modeling type of thing, so we know that the amino acid which is actively present in the active site.
Yeah.
So
we can do that. We can really do that. There's you might have heard of the software SwissProt. Okay. The Swiss modeling, the secondary Swiss modeling. Go into the software. It's very easy. For every protein, okay, every amino acid, it goes with an information. Whether it's it will take an alpha helix or beta bends or beta turns or beta seeds, etc. So every protein has got its quality, and it depends upon what is previous and what is next. And then you can go for the modeling. Most of the people, most of these is in India, they're doing, but they're not looking into this minute thing. If they do the minute thing, they can find the answer. This is a good question. You have to look at the minute thing, and then this one will become a big change. That will be do that.
Okay, thank you very much, sir.
Okay. Yeah, I see that there was Dr. Lakshmi was asking, sir, can you please share the PPT of this? Yes, I will pass to your organizer through email, and your organizer will share the slides. That's possible. I will send the PPT slide. The things, most of the slides were designed by myself, so you can use it, no problem. I don't mind.
Dear participants, any other questions?
I know that this is too much for 1-2 hours, but anyhow, it's this is what I want to give you. But when I send the slides, you can also read it later, and you can find out how this can be applicable.
Sir, shall we wind up the session?
Yes, go ahead if there's no more questions.
Sir, my last question is that, is there any age limit to join the PhD in your department under your guidance if I recommend about the student?
Yeah, there's no age limit. One has to pass either NET or our entrance examination. So our entrance examination is CSIR based. We gave CSIR based examinations for my department. We have 7 faculties, and we are looking for 4 more faculty, including bioinformatics. Each faculty has got 2 students at the moment. I would say they are saturated, I would say, but we can go up to 8 students. But anyhow, 2 is limit. But 4 more faculties will join, so the 8 more vacancies will arise in next month. So next entrance examination will be in August 3rd week. That's what I decided in the morning. I will talk to the authorities and we will do that. So there's no age limit. Only thing is that they need to pass either NET or our entrance examination.
Okay, sir.
Thank you, sir. On behalf of Dr. NGP Arts and Science College and ATAL FDP Organizing Committee, all deans, principal, HOD, staff members of Biotechnology Department, we thank you for this wonderful session, sir. Thank you, sir. I thank all the participants for your patience. Kindly join for the afternoon session at 2:15 p.m. Thank you.
Okay, thank you very much, Dr. Sarnia, and thank you very much, everyone. And I'm going to leave the meeting. Thank you. I wish you a nice day ahead and nice learning in the afternoon. Okay, bye-bye.
Thank you,
Yeah,
sir.
tomorrow.
See you on tomorrow,
Right.
sir.
Thank you very much.